Hello, welcome back to Biodesign Challenge Summit 2022. I'm Dan Grushkin, Executive Director. Welcome to day 2.
Hi, everyone. I'm Alex Kizalewski, BDC's Partnerships Director. Super happy to be here with Dan to welcome everyone. For those of you who didn't join us yesterday, just a quick explainer of what you're going to see with these presentations. We've asked each of the student teams to create a project video to explain the work they've done and their idea. We're going to play those project videos for you. They're about 5 to 10 minutes. And then we'll bring up the students live to the feed, and the judges will join us for Q&A with the students, ask questions, give feedback, stuff like that. And then we'll move on to the next team. So we hope you enjoy all of these projects for the day. One final reminder from me, we are doing a community choice prize again. So you'll have the chance to vote for your favorite project, or the one you think should win the community choice prize. You can vote via the link on our BDC Summit event page. We ask you to only vote once. All right, over to you, Dan.
Thank you, Alex. Yesterday, we saw a number of really fantastic projects. We're going to see a full day of more fantastic projects. This morning, we're doing something a little bit different. We're going to be showing the first 2 teams will be finalists for the Ginkgo Prize for Biological Futures. We have Grace Chuang and Alex Ting with us, who will be joining in the judging of those teams. Those teams happen to be both eligible for the grand prize and also this special track prize. So why don't we get started? We'll start with KO University, and looking forward to spending the rest of the day with you. Let's roll it.
Once you step foot into Japan, you'll realize that you won't be able to walk down the street without coming across at least one vending machine. At slightly over 5 million nationwide, Japan has the highest density of vending machines in the world. According to the Japan Vending Machine Manufacturers Association, there's about 1 vending machine for every 23 people. Japanese vending machines sell all types of soft drinks, coffee, tea, cigarettes, candy, soup, and even sake and beer. While these machines provide incredible convenience and fun, we can't neglect the fact that the amount of plastic waste generated from vending machines is extreme, contributing to the salient marine pollution problem in Japan. Further, these machines run at 24/7, using 7 to 14 kilowatt hours a day, even when no one is around. With the concept of the post-anthropocentric vending machines in Japan, we tried to encapsulate the idea of reshaping a contemporary Japanese phenomenon into a future bioplatform. The envisioned concept explores the near and predictable future of essential consumption in Japan and the speculative story of surviving one era and transitioning into the next. Our initial vision for a vending machine as a contemporary bioplatform heavily relies on food fermentation for bioenergy production. Long before the existence of modern packed drinks and snacks, people in Japan used to make fermented food and beverages that were served for daily consumption. We investigated how different dominant crops in Japan, like rice and soybeans, are fermented in order to propose a self-sustained system that can produce natto, amazake, sake, and other fermented food and beverages. To support our concept, we also explored the bioelectrochemical process of microbial fuel cells as a green technology that can produce electricity by using the electrons derived from biochemical reactions catalyzed by bacteria. Hako 25 is a vending machine system that consists of 2 different fermentation containers. One is used for soybean fermentation, and the other is used for rice fermentation. The fermentation containers are connected with the production containers, where the finished product is stored. From production containers, the finished product is dispensed into a consumption container. From the upper portion of the fermentation container, there are separate chambers into which the biogas is released, and from there, another pair of pipes extracts the biogas into the microbial fuel cells. Then, using turbines, the biogas would produce electrical energy that can be used to empower a single functional process, such as temperature control of the fermentation and production containers. Speculating the further development of the vending machine as a bioplatform in the next 5 years, we propose a system that could be fully self-sustained. We would try to expand the production of bioenergy with the implementation of additional microbial fuel cells that will use rainwater and mix it with excess fermented waste, compost, and soil to create the necessary mud. With the biorecalibration of the vending machine and the biotechnological advancements in the very near future, we could achieve this goal of a self-sustaining system and proceed with our next objective based on the multi-species narrative. In this world that is naturally and technologically intertwined, HACO 25 is long since separated from human-centered production, serving as an energy source for diverse living beings. We can already imagine a different form of nourishment and transaction, where the human takes the role of a resource rather than just a consumer. HACO 25 in the 22nd century is an interactive symbiotic organism that is a constant collaboration between species, ecosystems, and technologies. We envision the vending machine as a decentralized matrix of self-functional lab-like systems. Extracting energy from plant roots and mycelium to support itself, the system is also enveloped with a tangible membrane that collects human sweat through touch. Afterward, the healthy microbes from the sweat are separated and combined with microorganisms that initiate plant growth to fabricate a nourishing product in the form of a jelly. The jelly, which will be consumed through touch and skin by both humans and animals, will initiate the growth of edible plant-based body extensions that can be consumed and further be used by birds and insects as a temporary habitat. If we are able to realize the production of the vending machine based on fermentation, supported by bioenergy, this project can become an incredible opportunity to use citizen science methodologies and establish participatory research with the local communities in Japan. We can engage the elderly and local communities that have experience with soybeans and rice fermentation and directly include them in the process of research and production of the machine to keep the local culture alive. We will also include local farmers that will be the main providers of the crops. Humanity now exists in a new phase where we must move away from the idea of nature as something to be dominated and exploited and reconfigure how we work with nature. The proposal for a post-anthropocentric vending machine is not an aggressive, fear-based answer to the environmental issues, but rather a vision for a steady process of transparent evolution that will create a new kind of empathy for all species.
We're back with the KO students. Students, what a great project. Super cool to watch. Judges, I'm sure you have lots and lots of questions. Feel free to turn on your camera and audio and feel free to ask your questions. Alex, why don't you go first?
Hi, everyone. Thank you so much for this project. It's so cool. And I lived in Japan for 2 years myself. And the word jidohan baiki, which is the word for vending machine, is one I'll always remember because it's so fun to say. And they're everywhere. So yeah, this project really spoke to me. One of my questions that came up when I was thinking about this project was, I guess like the kind of like replacement of the vending machine with like, you know, kind of traditionally fermented products that are like so core to Japanese culture and cuisine. Like that's almost like a separate part of like Japanese diets. Like it almost doesn't replace what's currently like sold in vending machines. So I'm wondering like what is the audience for this, and do you see these kind of like phasing out like that type of food that's currently sold in vending machines, or is this something that's happened kind of simultaneously? Yeah, like who is the audience? Like, are you creating a new market? Or are you trying to kind of educate Japanese society into thinking like what's currently sold in vending machines like should kind of be phased out, like given the ecological and like health impacts?
So I think you touched on it a little bit right there. We do want to educate people because a big part of our project is engaging with the environment. And like you said, there is an established diet that is distributed through existing vending machines, but a lot of that is wrapped in plastics, plastic bottles, a lot of plastic. And if you lived in Japan for a while, you know there's just so much additional plastic waste that's produced. So in the short term, no, we don't expect to replace every single vending machine with fermented foods, but little by little, we do want to offer that as an option. And in fact, these sort of short-term events showing new things, new creative things that people don't expect, those kinds of things go over really well in Japan. So if we can just get people excited about, oh my goodness, here's one piece of Japanese culture mixing with another piece of Japanese culture, that's an exciting event. Then over time, we can use these sort of abandoned vending machines that are all over the place. You know, you can see things just sort of decommissioned, forgotten about. Use those, fill those up, and hopefully over time, like you said, shift the diet, shift the consumer habits over to a more sustainable way of being. This is what we envision.
Thank you.
Great. Grace, please go ahead.
Yeah, great presentation or video. Thank you so much for sharing your work with us. I wanted to talk or ask a little bit more about the science behind it. I'm curious, with the microbial fuel cell, how much electricity would the microbes have to generate in order to power this vending machine to make it like a truly sustainable operation?
So the really nice thing about fermentation is it's a natural process, right? It doesn't need that much additional energies, and the foods that we're providing actually don't require too much extra energy to make. So there's some fermented foods that need very specific, either high or low temperatures, but the foods and beverages we're suggesting actually are pretty much room temperature, maybe slight temperature controlled. And right now it is possible to get not a ton of energy from microbial fuel cells. You probably need a lot. And there are researchers around that have a lot of microbial fuel cells really trying to push the amount of energy and what they can do with it. And we're hoping that over time, like we said, in the next 5 years, by 2027, that technology progresses to the point where we can get that slight temperature control, just a couple degrees. Again, I don't think we'll need anything like 17 kilowatt hours or running 24/7. We just need that slight temperature control. We think it's definitely feasible within the next 5 years.
Awesome.
Right. Thank you for your question, Grace. Karen, you're up next. Please go ahead.
I was wondering if you had the chance to actually build a fuel cell and test out some of this design ideas you have.
So, would you like to take this one, Ivana?
Question: if we had the opportunity to test a microbial fuel cell, okay, because the sound was a little bad. We only explored how like the products that we suggest to be in the machine, like natto and rice, are fermented, and we explored the process of fermentation and how the biogas is released. But we didn't have the opportunity to explore or like test a microbial fuel cell, which is something that we want to explore furtherly with our project and try to build an actual prototype and contact with companies like Tokyo Gas that can support us and work on a real prototype that we can produce.
Great, thank you. Leslie, go ahead and turn on your camera if you have a question.
Hi, thank you very much. This was wonderful, really interesting. I have a question about the container that the food goes into. Can you tell us a little about that? The container that where people get the food in from the vending machine?
Oh, are you actually talking about the container that, like the fermentation chamber where the food is being fermented, or the district, like the plate, the little plate that
the plate? Like if I were to get some
right.
and I take it, tell us about the container that I take away.
Yeah, ideally we'd be using completely biodegradable sort of stuff. We use a lot of rice. The sake kasu is a byproduct of rice. If we could use sort of rice papers for that, that's what we'd like to do.
Okay. By the way, I was in Japan, and I was amazed at all the vending machines, and not one was vandalized. If you did that here in the U.S., I don't know.
They have those locked up.
That is one of the reasons why the vending machines are so common in Japan, because there is like a low rate of vandalism.
Thanks.
Great. Well, we're about out of time. Thank you, team. Thank you, judges, for your questions. We're going to move on now to Robeson High School. This is actually a fantastic collaboration of a bunch of different organizations to bring together a team. Robeson High School, Olive Futures, and Firsthand. Let's roll the video.
I can guess what you're thinking. How did the world get so broken? And what can you do to help? Well, that story begins many decades ago.
America. Brown mothers suffered when it came to bringing life into this world. This was especially true for African American women, who were 3 times more likely to die during childbirth than white women in the U.S. The reason for this is complex and deeply rooted in decades of prejudice and racism. This meant that people of color didn't have access to quality health care, and biased doctors and nurses meant that Black women were less likely to be taken seriously when they reported symptoms during pregnancy.
There were disparities in heart-related maternal deaths as well. Conditions like cardiomyopathy, pulmonary embolism, and hypertension were the main killers in pregnant Black women, and sepsis caused by infection was deadly too. The lack of diversity in clinical trials meant that medicines and treatments were rarely made to suit the unique needs of people of color, and a history of mishandling medical data even led to dangerous problems like algorithmic bias.
The maternal deaths among Black mothers continued to climb with no end in sight. This eventually created a world where it was almost safer to birth their children in any place but a hospital. Luckily though, a small but resilient community of doulas existed to help. Oh yes, the role of a doula is an ancient one. Long ago, women exclusively gave birth at home with their female family members to guide them. The development of hospital births only happened in the latest century. While midwives provided medical care, professional doulas were trained to provide emotional, informational, and physical support before, during, and after pregnancy.
Doulas offer continuous support to moms during the entire length of labor and beyond, compared to doctors and midwives who may not be as accessible or present. These doulas are intimately aware of their clients' needs and became increasingly important addition to a mother's birth team.
At the same time, new technologies in medicine were emerging. It wasn't long before everyday people started to understand what's called mRNA therapies and how to create them. This was partly due to a severe pandemic that challenged scientists to develop an mRNA-based vaccine in record time, speeding up research and development.
mRNA therapies were a truly disruptive innovation at the time, and you should understand how they work. mRNA is injected into the patient and delivers the information needed for the body to create its own medication. By engineering mRNA to carry instructions to cells, everything from disease-fighting proteins to antibodies could be produced. Pretty much, the patient's body is transformed into its own healing factory.
Back then, there were a few limitations and hurdles with this technology, mostly involving how the mRNA was delivered to the cells, but over time, solutions were developed. Overall, mRNA therapies are easily created, inexpensive to produce, and are scalable, and importantly, there is basically no risk of any information integrating into the genes.
As the field grew, we could see the potential of this technology to help the mothers we saw suffering every day. We were inspired.
One by one, we each discovered the doula community in our own way, and we saw how impactful they could be. Back then, there were already studies that showed that doula care improves pregnancy outcomes, and this is still true today.
As the years progressed, we learned so much from doulas, and it wasn't long before we founded Sister Sis. The goal was to create a community-owned organization that provides a variety of cutting-edge services, products, and information for Black childbearing women. Over time, the community grew to include mothers, fathers, doulas, midwives, nurses, and even a few trusted hospitals and research facilities.
Based on the knowledge we gathered over time, we were able to develop our own biotherapies to accelerate our efforts. We trained hundreds of doulas like yourself to use those biotherapies and even built a small lab at our headquarters where we conduct research and experiments. After a few years, the biodoula had been born.
Biodoulas not only provide informational, emotional, and spiritual support for Black women, but they also provide physical support in the form of mRNA therapies that are created and shared within our community.
Sister Sis has developed 2 mRNA therapies so far. One treats cardiovascular issues such as cardiomyopathy, and the other fights off infections such as bacterial vaginosis by programming cells to produce specific proteins. The process of developing a new mRNA therapy is simple. First, we sequence the mother's genes responsible for fighting off either cardiovascular diseases or infections.
Then, our researchers determined the specific code in the DNA that tells the cells to produce the protein that we're looking for. We then build and synthesize the mRNA in preparation for testing. Finally, we run an experiment and evaluate the results. When we find something that is both safe and effective, the mRNA is purified and enclosed in a lipid nanoparticle so that it can be ejected into the mother.
I think one of the things that I am most proud of is our unique method for building upon the contributions of our ancestors. Yes, we come from a long history of Black contributors to medicine like Henrietta Lacks, who were unethically siphoned for their biological information. Sister Sis is breaking this cycle by making sure that mothers who contribute to our cause are involved in the process of our research. When moms decide to share their DNA via a small blood sample, it is sequenced and stored in our system. At that point, it is only accessible to them and our most trusted researchers.
As time passes and we acquire more sequences from pregnant women of color, the treatment for our community continue to improve in a safe and transparent way.
You have made an important choice in becoming one of us, a birth justice warrior fighting for the basic human right to give birth in a way that is safe for the mother and the child. I am so proud of you that you are coming on this journey.
But before you receive the title of Biodoula, you must take part in your first ceremony. This is where myself and the other elders will give you and the next generation of biodoulas access to the research our community has gathered over the years. Value, appreciate, and protect this information always to ensure the survival and well-being of our people.
You will soon learn of our plans to become even more self-sufficient and to produce more life-saving biotherapies. In the future, you will help us develop personalized mRNA therapies and conduct research to create solutions for things like healthy hormone production in pregnant women. There is still so much work to be done.
Sister Sis was never meant to be the perfect solution for combating Black maternal mortality. Ideally, we would live in a world where people are treated equally and offered the same quality healthcare regardless of race or class. But The ripple effects of racism are even seen today. For now, Black mothers still need us, and we will continue to serve them. Help us to evolve what it means to be a doula, and show the world the potential of biotherapies in the hands of Black people. Have faith in all that you have learned in your training. You have done well. I now pass this responsibility on and gift you the title of Biodoula.
Judges, please turn on your video if you have questions. Grace, please start us off.
Yes. Thank you all for sharing that with us. I'm wondering if for the mRNA therapies, you could elaborate on that a bit more and also talk about if there is any data you found or research that supports that these mRNA therapies could help these women with their pregnancies.
Can you give us a second to talk within ourselves?
Yeah, of course.
Can Jessica?
While the students deliberate, judges, if you have questions, feel free to turn on your videos. First come, first serve. So whoever turns on their videos first will be the first to get to ask their questions. And then I would add that once you're done with your questions and the students have answered your question, feel free to turn your video off. The screen gets pretty crowded if we're all on at the same time. We're ready when you are.
All right. Well, so we were trying to find a way to tell the cell to produce these medicines for cardiovascular diseases and infections, and we came up with mRNA. And after talking with a bunch of other scientists, and they told us a lot more about it, we were able to come up with the plan to use mRNA therapies.
We mostly sort of, how should I put this? Kind of based it off of protein synthesis in a way, because, it was a good way to target specific infections and problems in pregnancy. Ones that are higher. That's the good wording.
Cool, thank you.
Great, thank you. Thank you, Grace, for your question. Alex, you're up next.
Hi, guys. Thanks so much for that. This is a really cool project idea. I love it. So I'm not from a science background. So from everything I know around social justice and from what I understand about doulas and friends who are going through that training process, everything about this project, I'm yes, let's make it happen. Let's do it. This is great. But we're also we're tackling huge kind of systemic problems here, around racism, prejudice, around the science, the whole science and technology of how it would work, and also the healthcare system. So I'm wondering, from your experience researching and learning in this project, what do you guys see as the biggest challenge to making this happen and making this a reality?
For our biggest challenge, I would say our biggest challenge would be growing the community, because obviously it wouldn't take a short amount of time because it's stretched on 70 years or so. So probably our biggest problem would be trying to reach out and get more members of the community to join and to get them to trust us. Because not everyone is just going to go hop on the board. Let's do this, let's do that, especially if it's health related and pregnancy related within people.
Great. Thank you, Alex. Emma, you have a question as well. Please go ahead.
Yeah, it's somewhat related. I loved the framing of the video that was recruitment. It's kind of addressing the challenge that you're describing. I like that it had kind of a recruitment related feel, like come join us on this journey. And one of the questions I had was about whether you all know about how many doulas exist, and then, in this country or in whatever context you are trying to scale this to. And then from that many doulas, how many would you need for us to even see, to recruit, for us to even see a dent? I really appreciated the acknowledgement of we're not going to solve the whole issue. There are a lot of other issues, but within what we can control, how many do you think would help to kind of shift the problem that you see?
We didn't really focus on that part. We didn't look into how many doulas we would need, but I'm sure it would be like a lot, over time as we grow.
So I would say, we would usually start off as a small number, just to pull people in. But the doulas have to be professional though, so they could get their point across and let it show that they were effective in what they were doing and how they did it. So we can sort of advertise our community to other people, other pregnant women, to grow the community.
Great.
I'll add on to that. It's the doulas are a very small community. It's very few of them, but there's a good amount of them. But with the doulas, there are enough for them to help support mothers during physical and mental when it comes giving birth, and helping them giving birth during the process.
Thank you, students. Thank you, Emma, for your question. Judges, are there any other questions? If not, I'll ask one. You had a really nice timeline showing how things might progress over the next 70, 50 plus years. Can you talk a little bit about how you chose that timeline? How did you pick your years and that progression?
Because it was speculative, we were we gotta drive this into the future. And because, in the future, things could either go uphill or downhill. So we chose for downhill, whereas, as in the video, Black women and other pregnant people of color women weren't taken as seriously as they were supposed to. So they chose to take it a matter of in their own hands to solve the problem themselves.
Great. Unfortunately, I'm sorry, Sukanya, we're out of time. We're going to go on to the next video. Thank you, Sister Sis. The next video is for University of Calgary.
I can guess what you're thinking.
Hi everyone, I'm Ashley, and with me today are Julie and Anna, and we're Team 5, Porifera. There's been a growing concern over the production of microplastics, which are plastic particles less than 5 millimeters, and their effect on sea life and human life. We first looked into issues we currently face, specifically products that are affecting our environmental footprint. As a group of 3 females, we quickly identified many issues with current feminine hygiene products, such as the lack of biodegradable materials, period poverty, and single-use products. We started out with a landscape survey to research popular products on the market, raw material composition, effects on the environment, and opportunities for improvement. From there, we identified pads as the least environmentally friendly product and narrowed down our topic to developing an eco-friendly menstrual pad. Over the years, many reusable menstrual pad alternatives have been developed, such as period-proof underwear, shorts, and swimwear. But there are a limited number of single-use alternatives that can be disposed of sustainably and safely. Currently, menstrual pads are made up of approximately 90% plastic and take up to 500 to 800 years to break down once disposed. The Porifera design is an original and innovative bio-inspired eco-friendly menstrual pad. It consists of 6 different layers, each inspired by various organism functions found in nature.
The top layer is comprised of a blended fabric of banana and bamboo fibers. Utilizing banana fiber will help to divert billions of tons of waste from banana tree stems, which are thrown out each year. The banana fiber woven with bamboo will create a soft, antimicrobial top layer for our eco-friendly menstrual pad.
Under the top layer, we will have an absorbent core inspired by sea sponges. This layer will absorb, filter, and expel liquids flowing through the product.
Under the absorbent core, we will have an antimicrobial layer inspired by the cicada. This layer will mimic the nanocone structure on cicada wings, which destroy bacterial cells upon contact. Next, under the antimicrobial layer, we will have a bottom layer made of a bioplastic which mimics the plaster bees' nests. This layer will ensure our product is leak-proof.
The adhesive layer mimics millions of small hair-like structures called setae on a gecko's foot. These microstructured fibers create large surface areas for adhesion via van der Waals forces. Next, the packaging will be made of recycled paper. This uses less energy, water, and produces lower carbon emissions than the manufacturing of non-recycled paper, and at the same time it reduces the amount of waste entering the landfill. Lastly, the menstrual pad will mimic the folding pattern of an earwig wing. This allows the pad to be folded into a compact shape approximately 1/5 of the original surface area for easy storage.
By looking to nature, we have been able to create a product that does not use harmful plastics. These materials allow our product to biodegrade at a faster rate when disposed of properly or improperly. In low and middle income countries where disposal bins are less accessible, the antimicrobial properties in our product still allow women to dispose of these products safely and protect themselves and others from any infections. This bio-inspired design effectively addresses 3 major issues, namely period poverty, sustainability, and public health. This project also most closely relates to addressing 5 Sustainable Development Goals. These include no poverty, good health and well-being, gender equality, clean water and sanitation, and responsible consumption and production. The entire life cycle of the menstrual pad was taken into consideration. This product will take 5 to 10 years to biodegrade versus traditional menstrual products which take 500 to 800 years to break down into harmful microplastics. Our eco-friendly menstrual pad will not change the living environment negatively. Period poverty is an evident problem globally. By offering an eco-friendly alternative to current market solutions, we are aiming to change the living conditions for individuals positively. We have chosen an absorbent material that is capable of regenerating when harvested. Local sea sponge farming has a faster generation time compared to harvesting from the ocean. Thus, our company will prioritize small-scale farming. This industry will also create more jobs for local communities, improving the living conditions of many individuals. Likewise, reusing banana fibers from the banana tree as our top layer allows these banana fibers to serve another function and diverts more than 1 billion tons of banana tree stems from entering the landfill each year. We conducted an in-depth literature review to explore sustainable fabrics with desirable qualities like high absorption indices and antimicrobial properties. This literature review was also performed to understand the landscape of existing feminine hygiene products. Next steps involve experimenting with building a working prototype with these bio-inspired materials and exploring methods to manufacture at an industry scale. Another next step would be to discuss with the banana industry on the best way to divert banana fibers to porifera manufacturing facilities instead of landfills, which they normally end up in. We recognize that the first prototype or model may be expensive due to being the first of its kind to incorporate new materials and technologies. In further iterations of the product, we will also strive to find economical ways to lower cost and increase accessibility. Moreover, this project recognizes its potential application to other industries, such as the possibility for eco-friendly diapers and adult incontinence products. The Porifera eco-friendly menstrual pad offers an innovative solution to billions of women across the globe.
Back with the students from Calgary. Thank you, students, for being with us. Judges, go ahead and turn on your video if you have any questions. Oh, that was a toss-up. I'm going to give it to Carolina first. And Ellie, you're on deck.
Thank you, Dan. Thank you, Porifera, for a wonderful presentation. I have a question in regards to the carbon footprint. As you mentioned, you're using, it's 6 layers, and you're using different agricultural waste. Where does this waste coming from, and where is it going to? And my second part of the question, as you mentioned at the end, this has a connotation with the price, because with your SDGs, you were mentioning poverty. So the price is definitely something that is going to be relevant for the development of your pad. So how do you envision lowering the price for this pad? Thank you.
Regarding the first part of your question, so we're planning for the top layer to divert waste from the banana industry. So right now there's billions of tons of banana tree fiber that just ends up in the landfill after the tree serves its purpose. So with regards to like the economical or carbon footprint of that, it would require diverting waste from those facilities to our facilities for manufacturing. But in hopes of in using that, we're diverting waste from the landfill itself. And then when our product eventually reaches a landfill, it would be a lower carbon footprint because it would be able to be biodegradable. And then Ashley, I don't know if you wanted to touch on the second part of that question.
Sorry, my connection's a little spotty. Could you repeat the second question?
Sure. It has to do with the price point. As you're doing your research, you're looking into continuously looking for a form of creating this pad at a lower price point, because that's one of your SDGs. So how do you envision lowering the price point of a pad that has 6 layers?
Right. So right now, it would be a little more expensive just because some of the layers have nanostructures. So manufacturing that would be a little more, but that's something that we would look into the future to kind of decrease that price, and also taking waste from different, like the banana fibers and stuff, that would help decrease the price as well.
Yeah, so we recognize that there is some more innovative technologies, but as we, grow and develop in that, I think definitely future iterations of the project, we'd be able to save costs or figure out how to make them in a more cost-friendly manner.
Thank you.
Allie, please go ahead.
Yeah, thank you. Team Calgary. It's a very valid and an important problem. It's kind of crazy that we're still using these kind of products with the knowledge that they're so destructive and have such a long-lasting impact on the environment. I guess my question is more around any prototyping that you've done in a physical sense. I know that you listed the 6 layers, you explained how they were inspired, or the different creatures and materials that they would bring in to the equation, but I didn't see any evidence of actual physical prototyping and combining the layers to understand how they work well together and how this might be manufactured.
Yeah, unfortunately, we didn't have a chance to prototype. In our landscape survey, we did research a lot of existing solutions, and in our material survey, just trying to figure out what was feasible or able to be produced with desirable qualities. So in our survey, for example, like the absorbent core of our menstrual pad is a sea sponge, and there are existing products, like menstrual products that utilize a sea sponge. So that was a really great confidence builder in using that layer, because it is already used, just not very common in North America.
Yeah, I thought it was interesting as well that you talked about the topic of the packaging and the way that the product would be folded in order to maximize, the amount of space or minimize the amount of space that it took up. Did you also do any experiments with regards to that, or is that just an assumption?
In our class, we studied the different folding patterns, and we did a folding exercise just with some paper. But that was also one of the things, is like trying to keep it smaller to maximize the amount of stuff we can ship in the box. And then also just the concealability of it.
Thank you very much.
Thank you, Allie. Judges, if you have any questions, feel free to turn on your videos. If not, we will move on to the next project. Thank you, students from Kent State. Enjoy your trip wherever you're heading. We'll now move on. I'm sorry, University of Calgary. We'll now move on to Kent State University. Thank you again.
Phragmites are rapidly growing reed grass with extensive below-ground rhizome structures that cause sedimentation buildup of untreated stormwater. They typically arise along man-made high-moisture, nitrogen-rich areas such as highways and construction areas. Wetlands invaded by Phragmites australis have 3 times fewer native species than untouched wetlands. To gain a better understanding of the impact that invasive species have on our environment, we contacted Rebecca Berryhill, grounds manager for Kent State University.
Vast monocultures and pushing out our native vegetation, it's so detrimental, because then, not only are plants affected, our animals are affected, and it just moves up the circle of life, throws everything off.
Textiles are traditionally sourced from natural fiber. However, in recent times, they have been replaced by rapidly produced synthetic, non-biodegradable fabrics. Could we use Phragmites as a source of a useful textile technology while simultaneously helping to manage its population? Introducing Innovasive.
Our ideation process began with a multidisciplinary approach, as we were coming in with a variety of backgrounds such as biomedical engineering, conservation biology, urban planning, fashion, and architecture. We identified key issues in these fields that we were particularly interested in, including an overabundance of invasive species, unsustainable building practices, and extreme waste in both the fashion and construction industries. We believed there was an exciting opportunity somewhere at the intersection of all these interests. We also drew inspiration from existing precedents in both nature and the built environment, such as how pine cones react to moisture, as well as kinetic clothing and kinetic buildings, including moisture-responsive workout suits by MIT Media Lab's team Biologic and wood facades by University of Stuttgart architecture professor Akim Menges.
These projects, however, depend on synthetic materials. According to the Textile Exchange, a leader in creating sustainable industry standards, the majority of cellulose products are largely extracted from wood pulp and contribute to deforestation and pollution. Our goal is to develop a textile technology engaging in the naturally reactive properties of cellulose sourced from abundant and repurposed invasive species while being cost-effective, efficient, and biodegradable.
To provide a solution for the gradual eradication of invasive species with a sustainable mindset, our proposal focuses on repurposing the waste products from the Phragmites extraction process by developing a water-reactive biomaterial, a hydrogel that originates from the Phragmites. This hydrogel could then be coated onto cut samples on plant-based fabrics.
In order to create the desired textile technology, the team began to study hydrogels as a potential actionable coating to be applied on the fabric. A hydrogel consists of a matrix of hydrophilic polymers that can swell and absorb water. It is important to note that this action is carried out without dissolving the material, with it staying intact as it expands and contracts. This expansion is repeatable as the macrocellular polymers absorb and release water. The team was led to a biopolymer known as cellulose, which is the most abundant naturally occurring polymer of glucose, found as the main constituent of plants and natural fibers such as cotton and linen. It is possible to use cellulose as a raw material to form a cellulose-based hydrogel.
In the beginning stages of creativity, we envisioned a product created entirely of Phragmites-based fabric in addition to our hydrogel. Phragmites have tough, bast fibers that are difficult to separate from the plant material.
To obtain the hydrogel, it was necessary to first extract cellulose from the Phragmites. The stalks were oven-dried and ground into a fine powder. Pure cellulose was extracted from the powder, and then this cellulose was converted into a derivative, carboxymethylcellulose, then cross-linked with 10% citric acid to form a stable hydrogel. Studies have shown that by converting cellulose into a more useful derivative before creating a hydrogel, it exhibits superabsorbent properties.
Hydrogel integration was conducted on various natural textiles including organza, cotton, canvas, and hemp. A series of 3 various types of cut patterns were designed and laser cut into the fabrics to determine the best cut geometry to allow range of movement with flaps. When the hydrogel is integrated with the swatches, the hydrogel can lower and lift flaps with the presence and absence of liquid. A pipette was used for precise application onto the swatches in a thickness of about 5 millimeters. In addition to total flap coverage with hydrogel, research suggests coating the fabric along the material's bias grain allows for more movement. The coated swatches were subsequently placed into an oven for 24 hours where the hydrogel could finish curing, then suspended vertically. Flap reactivity was assessed by wetting the swatches with a spray bottle. After 24 hours, the swatch flaps were analyzed to determine range of movement, and the hydration was repeated 4 times. Once dried, the flaps were measured to determine the hydrogel's effectiveness as a responsive textile technology. Analysis of the flap movement showed that when compared to a commercial carboxymethylcellulose hydrogel, our Phragmites-based hydrogel exhibited a greater distance of movement for both full coating and coating in stripes along the bias grain. The bias grain coating showed superior movement compared to the full flap coverage. Thus, it can be assumed that a Phragmites-based carboxymethylcellulose hydrogel fabric coating is capable of moisture reactivity when coated onto natural fabrics. Furthermore, of the various natural fibers chosen, hemp and cotton exhibited the most reactivity when coated along the bias grain of the fabric. To further test the hydrogel's viability as a fabric coating, its fireproofing properties were analyzed. In a lighting test, the Phragmites-based hydrogel burned at a slower rate compared to paper. As a soil amendment, the hydrogel aided in the germination of seeds. Over a period of 33 days, we observed the soil amended with the Phragmites-based hydrogel had a greater mean plant height when compared to its commercial hydrogel and plain soil counterparts.
The method of creating the hydrogel as a reactive textile coating aligns itself with the cradle-to-cradle mindset. We are mainly focused on the idea of a circular economy, utilizing plant-based materials as a source to solve issues with unsustainable construction materials while mitigating adverse environmental impacts. Compared to other hydrogels, such as polyacrylamide, our carboxymethylcellulose-based hydrogel is environmentally friendly and biodegradable. Invasive species pose a massive threat to businesses. Companies and organizations such as Kent State are paying large sums of money to have them removed. Innovasive would use this money to help offset the cost of extraction of the plant material. Here, the money spent is not just for eradication alone, but aids in producing a useful end product. Naturally and locally sourced plant species in conjunction with locally sourced labor for extraction reduces the overall carbon footprint of the production process. We speculate this model could be extended to encompass other invasive species as a control method to help eradicate the non-native specimens.
We are hopeful that our product, Phragmites-based hydrogel, can be utilized in future innovations in the construction field, architectural industry, and conservation ecology efforts. Our research suggested that different cellulose derivatives could be used to make a hydrogel to obtain determinate mechanical properties. such as thermal or pH reactivity and antimicrobial properties. Coating or hydrogel on the natural fabrics led to speculation applications. A Phragmites-based hydrogel with textiles assemblies could constitute an autonomous connected device. A coating for geotextiles that could give moisture for seeds to grow, or absorb the excess of water in flooded areas to gradually release it and moisturize the soil in dry seasons, which will require further testings. The sourcing of our product helps eradicate invasive species, while its biodegradability allows it to become part of a circular economy.
We are Innovasive, sharing the hope of creating a more sustainable and circular economy. We envision a landscape that utilizes naturally sourced technologies to generate inherently reactive materials.
Thank you, Team Innovasive. Excellent project. Judges, go for it. Turn on your videos. Carolina, first to the punch.
I really enjoyed your project. I think it has a lot of legs, and my question has to do with that. Did you envision or look into other byproducts other than the hydrogel? And at the end, what is the purpose of the hydrogel?
Sure. So we looked into multiple different venues for this. We initially looked into making a fabric out of Phragmites. However, that progress turned to be slow, so we turned to just testing the properties of our successful prototype of the hydrogel, and we are looking to coat it onto fabrics to give the fabrics different properties. And
Yeah, so as mentioned, we originally, in addition to extracting the cellulose for the hydrogel, we looked at creating an actual fabric sourced from the Phragmites. We are in talking with Praxis, which is a natural fiber company in Cleveland. So we look to work with them in the future to source a natural textile. And we were really seeing the hydrogel as an environmental, a coating on the fabric that could react to different environmental stimuli and help a textile, if it was integrated into architecture, to the built environment, to adapt to environment passively and help thermal control. So our hydrogel specifically reacted to moisture. We were thinking it could be used in applications such as rain shades or even help in flood control. But there's also in steps along the way in forming the derivatives from cellulose that we then take onto the hydrogel. You can fine tune that chemically to help the hydrogel react to other stimuli such as thermal response, pH response. So our project focused on moisture specifically, but we see a broad range of sort of applications that we could fine tune in the chemical process and obtain through our hydrogels and then coat on natural textiles.
Thank you.
Thank you, Carolina. Ali, please go ahead.
Yeah, I live in wonderful Costa Rica, which is incredibly humid, and humidity is literally a daily problem. Our shoes get destroyed, our clothes get destroyed, all our electronics in the lab are constantly under surveillance because, you know, the chances that they get moldy and everything is very, very high. I thought it was a very interesting use or repurpose of an invasive species, and I thought it was a great presentation. You answered a lot of my questions as you went along. I suppose one question that I wasn't quite clear on was, is this something that you imagine would be applied as part of the manufacturing process of the fabric itself, or do you imagine that this gel would be a standalone product that you could apply to existing materials. And then I have a second part to the question, which is you talked a lot about the business of fashion and also about construction and interiors for homes. And I can see how this gel, as it currently is very kind of crackly and quite hard. So I can imagine it as more of a construction material rather than something which would apply to, for example, clothing or something that you might wear upon your skin. So two things there.
Sorry, could you repeat the first part of the question?
Yeah, it was whether you imagine this gel would be a standalone product. So is it something that people could apply to existing materials, or is it something that or something that would need to be integrated into the fabric as part of the manufacturing process?
Right. So far in our prototyping, it was integrated onto the fabric as part of the manufacturing process. But aside from that, we sort of speculated, well, since the hydrogel industry is a large industry and the textile industry is a large industry, like we sort of have two routes we could go with both of our products, the sustainably sourced textile and the hydrogel. And we think it's interesting, the hydrogel itself is a more sustainable alternative to specific hydrogels that are on the market. And so in addition to being integrated onto the fabric, it could also sort of take its own path and be integrated on other fabrics or be used in other hydrogel applications altogether. So current prototyping is with the textile, but we envision it going having multiple uses.
Great. We're out of time. I'm sorry, Emma. Tyler, Celia, Alyssa, thank you so much for your time. Thank you for this great project. We're going to move on now to Franklin and Marshall. Let's roll the video.
Lancaster, Pennsylvania is well known for having some of the worst air quality in the nation, 6th worst to be exact. This is a result of its flatness, geographic location, and land use. While this is an alarming issue for the general well-being of residents in Lancaster County, it has become ever more important since 2020. With the rise of COVID-19, studies have found that poor air quality is closely linked to COVID-19 death rate. A Harvard study demonstrated that an increase in PM 2.5 of just 1 microgram per meters cubed is associated with an 8% increase in COVID-19 death rates. Therefore, our group sought to find a way to tackle this issue in a sustainable way so as to not aggravate the issue of air pollution.
My name is Alekia, and I'm a senior here at Franklin Marshall College, located in Lancaster, Pennsylvania. Growing up, I was a bit more prone to getting sick than everyone else. A normal cold that would take a person a couple of days to get over took me twice the amount of time. It got better as I got older, but then got a lot worse after college started. My first year and a half here, I got sick so much that I was missing class way more than I had ever had before. I went to a number of specialists, like the ENT and the allergist, and I found out that I was prone to certain types of respiratory illnesses. I was able to get a vaccine that has helped me tremendously, and now I have an air purifier on in my room at all times. I wasn't aware about how substantial Lancaster's air quality issue was until my first year here. I also wasn't aware that it could contribute to weakening your immune system.
Although the main source of air pollution in Lancaster is industrial and agricultural pollution, the effects are seen at a residential level, so that's where our product comes in. We designed this mycelium air filter to incorporate activated charcoal around an enclosed fan. The fan pulls air into the device and forces it through the porous mycelium. Through the process of adsorption, the activated charcoal collects contaminants, while mycelium acts as a structural support and additional service for filtration. In order to source our charcoal sustainably, we rely on woody landscaping waste and wood from the tree of heaven. Both of these resources are underutilized and have great potential for use in making charcoal. Most biowaste in this region specifically is used for compost and returned to the agricultural industry. But woody waste takes time and/or processing to compost. The tree of heaven is an aggressively invasive species that also acts as a host to the spotted lanternfly that is so destructive. Mycelium in the form we will be using is an inert fungus. Using simple ingredients like flour and water, a mycelium starter can be used to grow a structure with minimal processing. This poses a benefit compared to other sustainable filters on the market that use bioplastics that are heavily processed. We do not currently have a sustainable fan system. However, there is potential to use recycled electronics and renewable energy to run the system.
Welcome back, everyone. We're here with the students from Franklin and Marshall. We already have Leslie up for a question. So Leslie, take it away.
Hi, thank you. I really love your project. So thanks a lot. I mean, it's really interesting. I was wondering about the charcoal creating process, if that creates any kind of pollutants or CO2, or did you guys investigate that?
The charcoal creating process, we didn't really go too deep into that, but as far as I think, it was basically you take charcoal, you grind it up, you mix it with lemon juice, and you cook it for a while, and there you have your activated carbon.
Yeah, which is a great. It's really great for absorbing and adsorbing.
Mm-hmm.
Thank you.
Yep.
Thank you, Leslie. Judges, any other questions? It looks like, here we go. Carolina, please.
Hi, thank you for your presentation. So in regards to your process, I wanted to know a little bit more what opportunities and what failures did you find along the road that helped you towards the end product, because usually our failures, in my experience, are really conductive to making a better outcome of the process.
So we started with the idea of improve the air quality in Lancaster. So Lancaster is like a small city, and it has like a severe air pollution issue. It's like the sixth worst in the country. Like it's just such a small city, like with like really severe like air pollution problem. So we just came up with the idea of like building like an air purifier. You know, like to make an air purifiers usually like made by like a plastic. So like we want to make it more like sustainable with like material from natural environments. So like the material we use, like we have done a lot of like research on what material we should we should be using. So like we got the idea from like a mycelium building. So somebody like built like a building out of mycelium to absorb like pollution. Like we think, oh, we can like also like use mycelium to build like air purifier.
Just going off that, we prototyped a lot, and we faced a lot of challenges also because it is a new material. We'd never worked with this before. So, like things like shaping the material, or like how long to dry it so it doesn't crack, but it can be molded at the same time, and things like that. And just like how many filter layers to put and stuff like that. It was a challenge, and we prototyped it a lot.
Thank you.
Thank you, Carolina. Other judges, if you don't have questions, I have a couple. Why don't I start with, can you talk about what kind of testing you did compared to other types of air filters? I know there are a number on the market. And then can you talk a little bit about how this well, I'll start by saying this. It's surprising to me that Lancaster has such an air quality problem, considering it's relatively open landscape. It's quite agricultural. How does this solution fit into a constellation of other solutions to lower air pollution in the region?
So I'm gonna start off by answering the first question. So we actually use like a PM 2.5, PM 10 like meter to measure like the amount of like PM 2.5 in the air, like compared to like a normal air filter. Like however, the problem with like we haven't like completely solved yet is like we don't have like a strong enough like fan system to pull in the air. So like we purchased like a really strong fan system, but like by the time we finished the video, like the fan haven't arrived yet.
Got it. So then that might be the next step. In terms of solving the air pollution problem, can you talk a little bit about that? And then Grace, feel free to turn on your video.
I feel that our air purifier caters more to like residential use and not like a large scale, used for like large buildings and stuff like that. And our main inspiration was that a normal air purifier, like the production of a normal air purifier takes up so much plastic and like, acrylic fibers and stuff like that. And ours like is completely natural. And even the fan system, we would try and make it of like steel, because steel is like one of the most recycled substances and can be recycled again. But other than that, Lancaster has such bad air quality because it's first of all, it's flat and there's like a lot of agricultural waste and pollen and spores around. And I researched into this, and it said that it's downwind from larger cities. So the wind systems just carry pollutants into Lancaster, and it just settles down over here.
Got it. Grace, did you have a question?
Yeah, sorry. I need to rethink my question, so someone else can go first.
Great. Sorry, Jack, I didn't mean to cut you off. Go ahead.
So just really want to add on to that, because like Lancaster not only have like a air pollution problem, we also have like a problem with the invasive species such as like tree of heaven. So there's like a lot of like tree of heaven in the in our like school territory. There's like we have like over the football fields. There's our school like owns like a territory of like forest, and like tree of heaven have always been an issue there, just being invasive and like driving like native species into extinction. So like by making like charcoals out of like tree of heaven, we can like solve this issue.
Thanks, Jack. Grace, I think this is your moment. If you've rephrased your question, I would go for it now. We only have a couple of minutes left.
Sure. I'm curious about like if you could talk a little bit more about the reasoning behind choosing charcoal and mycelium versus, say, other forms of bioremediation, for example, like a microbe binding to a protein that could help with air contamination or something like that.
Like the basic idea is like the material like we're trying to use are like accessible in our local community. That's the main issue. Like both mycelium and charcoal, like made of like tree of heaven, is all like pretty accessible.
Got it.
Also, just going off that, mycelium grows on agricultural waste, and since Lancaster is so agriculturally dominated, agricultural waste is like really easy to get. And instead of like sending it to landfills or like burning it, this is a great way to like reuse it and like, make stuff out of it, which can be sturdy and which can be converted into like air purifiers or mycelium bricks or things like that.
Great. Thank you. Ali, please go ahead. This will be the last question. Oh, you're on mute.
Sorry, new to Zoom after 2 years. And my question is around how to visualize how much pollutants are being taken out of the air. I think when you have something which is a bit more industrial rather than biological, you can measure quite accurately, how much air is being purified and the quality of the air that you're putting out. Have you thought about how to make that information visual?
Yeah, we'll be thinking about like doing stuff like that in a video, like our air purifier is more like for like a household setting. So we were trying to reach like an industry level of like air purifier, like that level of like how much you can pull off the pollution. But like as I said, unfortunately, we don't have like a strong like fan system to support our idea. So that's kind of like, a pity.
Thank you very much.
Well, thank you both. We're going to now move to a 15-minute break. Thank you so much for your project. So we're going to take a 15-minute break. Folks at home, we're going to show some videos of former teams, as well as artists in our communities, and videos of sponsors as well. Please do join us back in 15. See you soon.
The universe in the natural world is the best designer. I don't think there's anyone that does color, shape, form, sound, light better than Mother Earth does. So I've always been in love and been inspired by the natural environment. Design is about solving problems, and sustainability is the biggest challenge of our century. Great designers love good challenges. And I think it's why we are making headway, is because I remember seeing the fire in their eyes when I said, we must take this on.
When we think about designing for circularity, it touches on a few different aspects: reducing waste and pollution, designing more efficiently from the inside out, designing products that last and grow with us through different life stages, and of course, trying to incorporate as much recycled content and bio-based materials into the product. The more we hold hands, the more impact we will have. And so it's really sort of getting in there and asking good questions and supporting each other. There's a lot of breaking process that happens, but it's also really exciting and very rewarding.
Our design team constantly is thinking about how our materials and processes can be developed to be recyclable, reusable, reformable, with minimal pollutive outcome. We want to help reshape the industry's dependence on making new materials and instead switch over to 100% reuse of waste materials that would otherwise end up in landfills and give them a second life.
We need to look at hardware, but also software and supply chain, and from how the materials are sourced to how the product is put together and how it's also taken apart, and what happens to the product after its life.
I see us as being at this inflection point of being able to innovate and create solutions that don't yet exist, that address more of the problems that we've never intended to create, but we hope we can fix in this next go-round. To me, a lot of this also attaches to the people we're listening to, the people we're partnering with to create those solutions, to create those products. I think the world overall is at a place and understanding that there can be a more equitable future for everyone, and that we can address those unintended consequences really from the start.
Sustainability is absolutely critical to our consumer hardware journey. And I think you can see it in some of our products. I mean, they when you look at them, you can tell that there was a diverse team that was thinking through them. And that's probably the biggest reward, is to be able to see that. I'll show it to my kids and they're like, oh, I really like this, I really like that, this is really cool, it doesn't look like the rest. Well, it doesn't look like the rest because it was built in a more inclusive way.
People think that corporations should be doing more in this space. And I think you've always said we need to make it a benefit for our customers. This can't just be like a good idea from a scientific perspective, but it has to really resonate with their heart.
We have been saving energy for the past decade with our Nest thermostat, and we wanted to allow more people to save energy with our new product. And not only are we saving more energy, but we were able to make it more sustainable at the same time, and without any compromise in beauty, in how it looks.
You don't have to compromise beauty or function to do what's right. So I look forward to discoveries, collaborations, creations, where we move forward and we secure that humanity will stay alive and that we take care of Mother Earth that created us.
Devin and I both have participated in the Biodesign Challenge with students from the Midwest in the past, and we've had very different approaches that our students have taken. And so I sent a bunch of questions to Devin, and he's prepared a couple of responses to those. And I think that he has a presentation that we're going to go through together.
Yep. All right, so can everyone see that?
Yes. Awesome, thank you so much. So one of the things that's really noticeable about Devin's students' works is that they really highly rely on the speculative. And so one of my questions initially was just, why practice speculative design when we face so many real world problems out there?
Okay, so I figured we could start with just a kind of quick defining of the terms. So, if we're talking about speculative design or speculative futures, the big question is, what actually is a speculative design, and how does that kind of differ from other types of design? And I think, as Andy mentioned, his classes use maybe something that might be called, utilitarian design. They're trying to solve real world problems. So how does that differ from speculative design? So this is kind of a conceptual framework. Anyone that kind of has been around speculative design for a little while has probably seen some version of this. This comes from Tony Dunn and Fiona Raby, who wrote the book on it, literally. And so over here on this side, we see, okay, utilitarian design. If we think of these as like dipoles, right? Utilitarian design is sort of affirmative, it's problem solving, it provides answers to big questions. It might be sort of what Nandita called like problem solving certain wicked problems and providing answers to those. Often it's in the service of industry, and so it's concerned with futures and design for production. Whereas, you know, on the opposite end of the spectrum, we've got something that is often labeled speculative design, where usually it's critical. Instead of trying to solve problems, it's looking for problems, and it's just simply asking questions. And so instead of being in the service of industry, it's looking at how can you make design for society. And, you know, there's a bunch of different sort of ways to categorize this. Often you'll hear like user design. I teach a class on user interface design and user experience design. And so getting students to really think about the individual person with a lived experience behind the user is kind of often a big challenge. So I think speculative design can really help with that. So, however, there's like other ways to explore the differences between utilitarian design and speculative design. And so I think the best way to sort of understand this is to talk about the future and to realize that there's not just one future, there's multiple futures. And so this diagram provides like an overview of like a richer language for discussing speculation, for discussing futures. So over here, we've got the present. And often, you know, the very first thing we think about when we're talking about futures or speculative designs, we think about kind of the movement from the present to the future. And so inside this sort of cone, right, that's everything that's scientifically possible. Outside over here, this is like magic. Inside here, this is kind of what is possible within our sort of physical laws of the universe. And then sort of a more narrow framework is what's plausible. So that's the stuff that's likely to sort of get done. And then you have the probable. So this is even more narrow. So, you know, you might think of the plausible as, okay, in theory, we could get rid of COVID in 2 weeks if everyone in the world just went inside their homes and quarantined for 2 weeks, and then it would be gone. But that's probably not going to happen. So this gives us a richer language to talk about kind of the variable futures that exist. And then over here, we have the preferable. And this is often sort of the framework where a lot of designers are working with. And so this kind of diagram was first presented in Speculative Futures, or at least that's where I came across it, from Tony Dunn and Fiona Raby. And it's called the PPP Futures diagram, first created by Stuart Candy. And so I have a slight addition to that. And so there's this other realm of different futures within design or within art that are contestable. So contestable futures are speculative futures that feature the abject, the grotesque, the profane, the undesirable, the horrible, the absurd. And in general, these are often imagined futures that we don't want to happen. As an entire population, we want to avoid these kind of futures. So I would contend that, you know, most design is speculative. But when we're talking about the term, quote unquote, speculative design, we're actually referring to contestable design. So even utilitarian design is inherently speculative. So when we think about the biodesign challenge that occurred, well, for the last 7 years, but, you know, definitely this past summer, all of those projects are proposing some sort of speculative future, but they're using a utilitarian outcome. So I would say most utilitarian design operates within the possible, but it sort of has this little donut hole where the contestable isn't really examined or looked at. And so I think most utilitarian biodesign projects probably, you know, seek to be in this preferable range. They're trying to solve real world problems. They're trying to fix a lot of the difficult situations that, we're currently in, which is, by all means, it's a very good and honorable sort of task to undertake. But I would say that most, quote unquote, speculative design projects end up actually operating in the realm of the contestable. And so this term, the contestable, I first came across it from, when I was in Australia pursuing my master's degree with Symbiotica, which is an art and science research laboratory in Perth, Western Australia. I first heard the term from Oron Katz, who's the director of Symbiotica. And so, the key thing here is often when we're talking about speculative design, we're really referring to futures that are maybe contestable.
Welcome back, everyone. Next up, we have California College of the Arts.
If Darwin were alive today, the insect world would delight and astound him with its impressive verification of his theories of the survival of the fittest. Under the stress of intensive chemical spraying, the weaker members of the insect populations are being weeded out. Now, in many areas and among many species, only the strong and fit remain to defy our efforts to control them. This was an excerpt from Richard Carson's revolutionary book, Silent Spring. Carson and her breathless fight against the environmental destruction caused by DDT, one of the most popular pesticides that emerged after World War II, forever changed how we thought about the use of chemicals as controlling agents against insects. In her paper called Our Oldest Enemy, Tanvi Dutta Gupta, biological scientist from Stanford, mentions that DDT embodied the victory of the West over America's enemies, and over insects and mosquitoes in particular. She also discussed that California has had a complicated history with mosquitoes. Malaria came to California early by hitchhiking on European settlers. It killed gold rush miners and devastated the local indigenous people. The history of mosquito nets, on the other hand, goes back to the old times, but nowadays they are mainly made out of plastics. Mosquito nets that are designed for protection against malaria are often treated with synthetic insecticides that are harmful to the environment. These chemically treated nets are 50% more effective than untreated ones, and their protection is 70% greater compared to no net at all. The fine mesh of these plastic nets are also used in the illegal bycatch of the fish in our oceans. It's been studied that these nets allow for extracting the smallest fish from the aquatic environments, which causes enormous problems in the food chain balance. In addition, the insecticides also kill the fish. In places like Nigeria, contraction of malaria still remains a fatal issue, and the distribution of these nets remains complicated and manipulated by the socio-economic factors. With the rise of the temperature due to the climate change, insect control will be the second most challenging issue that we have to face following population growth. And we are clearly still at war with mosquitoes. They trigger our insect to survive, but the chemicals we use against them have made them immune, poisoning our environment and ourselves in the process. What if we could replace the industrial plastic manufacturing of the mosquito nets with the local production of biomaterials, reusing food that goes to waste and replacing the synthetic insecticides with natural ones, all in one net? Research has shown that citrus peel contains organic chemicals that are able to repel adult mosquitoes. The compound D-limonene is found in citrus peel and lemon essential oil. This organic compound is a natural mosquito repellent. In addition, lemon scent has shown an ability to repel mosquitoes. Based on the evidence that mosquitoes dislike lemons, I decided to choose lemon waste as the starting point for biomaterial development. During the process, I dissected lemons in many different ways to familiarize myself with their anatomy and their physical properties. I also used their peel, zest, and pith in many experiments to make bioplastics and to test their resistance against mold. In my next step, I developed methods to make net patterns out of my most successful biomaterial samples. I made threads, connected them with knots, used weaving techniques, and played with liquid expansion on the surface. I remelted the failed materials and mixed them with new ones to create different colors. In this process, I discovered materials with new qualities such as stickiness, which seemed the proper physical quality for the mosquito net. The natural next step was making molds and dies to achieve precise and repeatable results. I 3D printed and CNC cut molds for casting, 3D printed dies that would punch holes into the semi-dried material, and overlaid 3D generated patterns to examine the moire effect. This stage of the project was the most challenging and resulted in many failures due to the desired thinness of the material, its rapid setting rate, and the small mesh count. I kept trying different punching methods using needles, nails, and straws, and finally an array of skinny straws made the most successful and promising sheet of material. This is the very beginning, and I can't wait to push this vision further by collaborating closely with engineers, chemists, and manufacturing experts to achieve a mesh count suitable for the protection against mosquitoes. I propose casting and punch pressing as manufacturing processes at this stage, but 3D printing and filament could be other viable additive process in future, substituting the material removal. During the process, I also realized that it's possible to develop a new bioplastic recipe using entirely lemon parts. Lemons already contain all the elements of a starch-based bioplastic recipe. Lemon peel will provide pectin, glycerin will be made from lemon oil, and lemon juice will act as the acidic component. Lastly, D-limonene will be embedded in the material as a mosquito repellent agent. With this purist approach, I want to argue that the replacement of plastics with bioplastics should not justify the excess use or extraction of other bio-based materials such as agar-agar. The material experimentation and development was an incredibly exciting process, and I learned so much from every step, mostly from my failures. Now, let's zoom back and take a close look at the synthesis of my resolution. The closed circular system starts with lemon-based bioplastics. This has two advantages: lower carbon emission compared to production of plastics, and removal of synthetic insecticides, replacing them with natural ones. Next, mesh patterns for the mosquito nets are created inspired by the shape of the mosquito wings, insect herbivory on lemon leaves, and the inner pulp structures of a lemon. In the next phase, I propose to make an outdoor tent with this textile, because tents are symbols of nomadic life and their origins goes back to the beginning of humankind. Historically, tent making has a strong relationship to the socio-economic factors of each region. As Philip Drew mentions in his book, New Tent Architecture, tents are ephemeral, mobile, flexible, sensual, and feminine. They can also be a solution to the rise of human need for mobility and survival due to climate change. At the end of the life cycle, the mosquito net is cut in pieces and burned. This ritual burning creates smoke, which mosquitoes detest, and releases lemon scent. The lemon ash from burning will be reused as pigment to make the new nets. I chose California as the initial location to test my concept and to iterate on prototypes. But I can't wait to expand this project to other regions in the world and to work closely with communities where mosquito blood transmitted diseases are a bigger issue and where people don't have easy access to these nets. Malaria is a climate problem. Distributing these nets shouldn't be a monopoly, and manufacturing them shouldn't be an ecological burden to the ecosystems. I want to create an economic system in which people have the power to grow lemon trees and to make their own lemon-based mosquito nets on local scales. The importance of mosquitoes in the environment is often overlooked by the other natural conservation efforts, such as saving beautiful forests and exotic frogs. But mosquitoes are essential for the health of the ecosystem. They pollinate flowers, and other animals like bats, fish, and birds rely on them as food. Eradicating mosquitoes at any ecological cost isn't feasible anymore. The aesthetics and visual aspects of this mosquito net aims at acknowledging their importance and their harmony in nature. This project is a small portion of a much larger debate about speciesism. I want to argue that we as species should see ourselves as only one part of the ecosystem. We can live more intimately with nature, co-evolve with insects, and protect ourselves and all the other components of life at the same time. We have to reimagine what we call nature, how we interact with it, and how we protect it. Thank you.
Welcome back. Thank you, Sarah, for a fantastic project. Really impressive prototyping. Judges, I'm sure you have lots of questions. Feel free to turn on your videos. Sukanya, please start us off.
Hi team, great presentation. So my question is,
what is the efficiency of your nets? So I know you said you tested them in California. So how well does it work?
Hi, thank you for your question. I think I'm still very at the prototyping of the net itself. And in terms of testing against mosquitoes, I think there's still a lot of work that needs to be done. Even my mesh count is not still small as it needs to be compatible with existing mosquito nets. So this is just an exciting moment to show that it's possible if I'm able to collaborate closely with chemists and scientists and other experts to be able to achieve a prototype that we can test it. And also in terms of understanding if mosquitoes are able to pass through the test, I think there are much more technical tests that needs to be done, which I didn't have access to.
Thank you.
Thank you. Leslie, please.
Hi. Are you the only one on this project?
Yes.
Oh, great. That's amazing. I wondered if you tested the lemon against commercial pesticides that are used that are are very effective but cause problems. Obviously, environmental problems. Can you talk a little bit about how effective it is in comparison?
Yeah, totally. I did a lot of research about synthetic repellents versus natural ones, and by definition, they're way less effective. But that's also the main point. We cannot put all these chemicals in the cost of just protecting us and causing all these other problems in the whole bigger system. So the natural repellents in general are around a third to a fourth of the efficiency of, like, let's say DEET or other chemicals that we use. But with the use of mosquito net that I'm proposing, we don't need to have that much of chemicals. And we can just replace the net that has lemon parts embedded in it. And I'm hoping that this would be a viable and reliable alternative.
Thank you.
Of course.
Thank you, Leslie. Carolina.
Congratulations on working on your own. I know it's a very a process that takes a lot. So you did a fantastic work. So I have a question. I'm from Colombia, and malaria is definitely an issue in the coast, in the Pacific coast. And one of the processes that they use is that they use a DDT and they spray communities that might have a big malaria issue. And what they found is that the communities hide at certain times during the day where there are more mosquitoes. But I think that within your process and your project, another area that you could look into would be in using it for clothing. I don't know if it was something that you envision, and if your background is fashion or textile.
No, it's not. But I think that's a good point. I finally, in the conceptual phase, I was very fixated on creating a mosquito net. But as I developed through making my prototypes and all the sheets I made, it can definitely have so many different uses and be embedded more in like the outdoor gear or outdoor clothing that we would wear as an alternative and not exclusively as a mosquito net. So right now I have the textile, so it can be used in so many different directions, I guess.
Yeah, and another part of my question would be that the mosquito nets usually cost between $2 to $4, $4 being the highest, and because usually they come to communities that cannot afford more expensive solutions. So in that respect, I know that bioplastics and the bio is a little bit more costly than a synthetic solution. So how would you compensate that price for communities that might be enhanced by your solution?
Totally. Yeah, that was something I was thinking from the beginning. And because I'm using lemon waste as the material, and I also proposed to create a brand new recipe only using lemon parts. So I wouldn't even need to use glycerol separately or acid or let's say agar. I can create all of those components for the recipe entirely made out of lemon waste. So I'm speculating that creating all of this pure approach would also decrease the cost moving forward. Obviously, the first prototypes would be more costly, but I think using entirely waste to generate a new material down the road would be profitable and lower the unit cost.
Thank you.
Thank you, Carolina, for your question. Emma, we're going to jump to Emma, and that'll be the last question. Sorry, Ali, we're actually out of time.
One of my questions was about cost. I'm glad that was answered. And then the other question is about durability. And did you do any tests kind of like thinking about the tents, being in the elements, or if they were on clothing, as you said, for a fabric? Did you have any sense of like kind of how long one of these would last?
Yeah, I think around 3 months I have landed on the material and I have been prototyping, and I did leave them in sun for so many hours. And I think right now they get a little bit sweaty, but not degrading, obviously. But I think I need to embed or tweak the recipe a little bit more towards water resistance and heat resistance if it will be used outdoor. But yeah, definitely those are the factors I would need to work so much more on to perfect the qualities. But right now it is very flexible, and it's almost like not easy to tear it apart, even with the holes in it. So I think that's an exciting start.
Great. Thank you, Sarah. Thank you, Emma, for your question. We'll move on now to Rutgers University. Let's roll it. Thank you, Sarah, again.
Thank you.
Hi, I'm James.
Hey, I'm Corey.
Hi, I'm Kaylee.
Hi, I'm Fedwa.
And we're the
micromelanin transducers of New Jersey.
Micromelanin Transducers of New Jersey proposes to view radioactive waste in the Garden State as a new energy source. Utilizing the ability of melanized fungi to convert radiation into chemical energy, this project envisions a world where our radioactive waste will power natural cycles of remediation, generate protective barriers, and even grow shelter. This project is inspired by our hands-on work with fungi and the Biodesign Challenge, which encourages designers to view our waste stream as our resource. The project was also inspired by our home state of New Jersey, which contains more Superfund cleanup sites than anywhere else in the US. Superfund sites are polluted locations in the United States requiring a long-term response to clean up hazardous material contaminations. However, according to the historian Howard Gillette, during World War II, these places of industry may have been the most productive sites on Earth. This productivity may have allowed the US, along with our allies, to win the fight against fascism in the middle of the last century. So these post-industrial spaces and the communities that house them deserve our thanks and our support. Our team researched the US Radium site in East Orange, New Jersey, a place that several of us live by that has famous history of radioactive contamination. Opened in 1917, the plant used extracts from radium ore to produce luminous paint for glow-in-the-dark watch dials. US Radium encouraged the young women working there to wet their paintbrushes on their lips. These workers suffered terrible diseases and filed suit. Among the most painful and disfiguring was radium jaw. Known as the Radium Girls, the publicity around the case helped establish occupational health and safety standards. Unfortunately, the company also buried its leftover ore at sites in the local community. This waste is dangerous for most living things because the ionizing radiation affects genetic material, DNA, which can damage cells and organs. In researching our project, the team made site visits and consulted with officials from the EPA. We learned that Superfund is a risk-based program that evaluates exposure pathways. One of their main concerns is exposure to radon gas. At the site, EPA investigated about 650 properties for radiological contamination and excavated soil from about 250 properties. The hazardous waste of this site was sent to a radioactive soil deposit in Utah. The area is now declared clean, but multiple sites in New Jersey and across the country are still contaminated.
The mycomelanin transducers seek to use this waste stream as an energy source to grow fungi superstructures. All fungi contain melanin. Just like in humans, it's used as a protective barrier against ultraviolet rays, especially in extreme environments like contaminated nuclear reactors. Similar to how plants utilize sunlight to grow, melanin is found in the cell walls of all fungi, which convert radiation into a usable energy source. This mushroom was found growing nearby the nuclear reactors in Chernobyl. This radiotrophic fungi survives by feeding on radiation with its high melanin content. Here are a few samples of wood ear mushrooms found naturally growing nearby the old Radium Corp factory site. This species holds some of the richest melanin content in its cell walls. We can utilize this fungus because it's native to the area. In our designated lab space, we experimented by growing different types of inoculants like reishi and oyster mushrooms. Each day we made sure to centrifuge our samples to make sure they were not stagnant. We also ordered 2 brown oyster mushroom startup kits to experiment with. We weighed out samples and grew the fungi to establish the rate of growth. We also experimented by making different substrate compositions while designing and 3D printing our own molds to grow into.
Our project seeks to utilize the amazing properties of this life form to remediate these spaces and to use our waste stream as a resource, and to potentially produce structures that look like this. In this building's construction, it would be a missed opportunity not to take advantage of what the building grows. In 2018, NASA proposed a design for developing on Mars to reduce costs of transporting materials to space. They mimic the symbiotic relationship between fungi and lichen by using cyanobacteria in addition to mycelium to produce oxygen for human occupants. Our design's external walls contain a system that replicates this symbiotic relationship. This is lichen under a microscope. You can see here that the cyanobacteria coexist symbiotically with the fungal hyphae that feeds off of the bark of a tree. The mycelium membrane exists in symbiosis with the cyanobacteria and can be supported with any flexible membrane. This can be made of wood, plastic, or hardened mycelium itself. The compost inoculated with mycelium spores fills the membranes in its basins, and over time, the fungal hyphae eats through the material. After about 2 weeks, we are left with a hardened and strong mycelial mass, which can be killed to prevent it from growing further by applying heat. This system is applied with an interior and exterior reinforced with the mycelium brace system. Underground and below our design, the mycelium grows into the surrounding soils, while digesting toxins and pollutants that exist in the ground of a Superfund site. The life cycle of the mycelium construction will also biodegrade and spread spores to restart every 20 years. This biodegradable construction replenishes the health of the city by consuming the city's waste and returning nutrients to the surrounding soils. Our building's design rendered at night looks like this.
While working on this project, our team considered the ethical implications of our proposals. Our concerns included the properties of radioactive fungi, that the sites may still not be safe enough for people, that our designs may encourage people to be near these sites, which could be dangerous, that because of these dangers, we were not allowed to work directly with radiation, so we were unable to study these processes. And also that societal inequalities may play out in the sites that are being remediated. So our group is recommending community actions like making micromelanin fungi balls that could be safely deposited at a distance into radioactive sites. In conclusion, we recognize that our project, either seen from a distance or utilized up close, allows us humans to actively engage these sites while expanding the habitat of these extraordinary life forms. The team is also excited to report that site administrators at the EPA are willing to allow us to present our ideas to the Research and Development Department for possible field testing.
Welcome back. I have the team from Rutgers with me. Judges, go ahead and turn on your cameras to ask some questions. Here we go. Carolina, please.
Hi, great presentation. Where in New Jersey are you located?
Carolina. So most of us are near the Newark area.
Okay.
Some members of our team are also located down near Camden.
Okay. So I live in New Jersey, so I pass by Newark all the time. And I didn't know that it had that impact with radioactivity. So that's very interesting to know that. In regards to the use of the oyster mushroom, I saw that you got 2 boxes. And I want to know, how was that experimentation? Because the oyster mushroom will take some time to develop, and you were running during your process and trying to get as much prototyping and experimentation during the semester. So I want to know a little bit more about that experimentation and your successes and things that might have gone wrong.
Well, thank you so much. That's such a great question. I'm going to pass it off to Corey.
Hi. Right. So we experimented with some of the brown oyster mushrooms, and we were actually able to grow our first sample pretty quickly within a couple days. And we look forward to growing our next sample to then send over to a Rutgers facility, which would be able to concentrate radiation in a controlled environment. And then before that, we worked a little bit with biomaterials like the mycelium kits from Ecovative and such. So we just really got used to the process of growing mushrooms, and this was our first time. So
thank you.
Thanks.
Karen, please go ahead.
I wanted to ask a question about the use of melanin. I feel like I've read some things that critique sort of using melanin in this way that feels exploited given the history. And so I just wanted to ask if you've thought any about like the societal implications of using melanin in this way.
Thank you. That's a really good question. And once again, I'll pass it off to Corey.
Yeah, so it's a great question. Like according to the experts that we spoke with, using these areas that have been contaminated. There's like a couple of them that have been remediated and cleaned up, but about like some ethical considerations would be that, like moving these mushrooms away from the area that they're grown in, we would be able to use these in different types of production. And also that it wouldn't be too big of a concern about, like overusing these type of sites. So, yeah.
Got it. I think I was asking a little bit more about just like the meaning that melanin has in terms of like race, ethnicity, and like skin color, and how if you've thought of that like in relation to your project.
Right. Like we didn't get too deep into that, but we noticed that obviously in animals and humans that contributes, that we have melanin and that mushrooms use melanin. But I think it's a great type of view to look at, since mushrooms are almost like human beings in a way. So I think further exploring that field would be interesting to present our ideas. Yeah.
Great. Thank you, Grace, for your question. Karen, we'll move on to you.
Great, thank you. Really enjoyed this presentation. Just curious, though, how we landed on mycelium mushrooms instead of some of the known microbes, bacteria, geobacter, for example, that are known to degrade radioactive waste. And did you explore any kind of symbiotic relationships, embedding microbes inside mycelium, or thinking about other environments like the ocean where we see a lot of radioactive contamination? So just would like to hear some more of your research that led you to the conclusion of suggesting mycelium.
So students, if you didn't hear Karen, you were cutting in and out a little bit. The question was, why did you choose mycelium as your material when there are other organisms out there that exhibit similar properties? Is that all right, Karen? Okay.
Right. So this idea came out of the research from a couple different experts. One we spoke with, John Dighton, and the research was on high melanin content found in fungi around the Chernobyl area. So that's really how our idea sparked. We figured out that mushrooms with high melanin content were growing in the area, and we weren't sure if this was from fungi which evolved there to form to the proper setting, or from like process of elimination. So really that's where our idea grew out of.
Emma, we'll close with you. Please go ahead and ask your question.
All right. I do some participatory design work in East Orange. We have a new project there with a Baptist church. So this was I started Googling. I was like, whoa, I didn't know this site was there. And I was nervous about how close it was and all that. So thank you for even sharing the information about the super fun site in East Orange. One of the questions I had as you were proposing a building on the site, were you able to talk to residents as experts of the area or understand how the site might have affected them locally?
Thank you. That's a really good question, and we'd love to visit sometime and see the area around. To answer that, I'm going to actually pass it to Fadwa.
We did not actually have the chance to go and visit. We did not have a lot of time, but there was some research, according to the people we talked to, that people like surrounding Superfund sites do tend to have a higher risk of getting, for example, cancer and other diseases, and of course have high risk of getting affected by the radiation from the Superfund sites.
Great. That's all we have time for. We're going to move on now to University of Michigan. Thank you, Rutgers. Thanks, team. Great project.
Greetings, and welcome to our presentation on our project, Jester, a biodigester service transforming our relationship to human-generated nutrients. Our team representatives are Whitney Brooks, Gabe Brower, Celeste Simons, and Jacob Yu.
Our project aims to address an issue of wasted potential, wasted nutrients, wasted energy, and wasted resources. Because of increasing climate instability, sewer systems are coming under increasing strain and failing to contain human waste. Our sewers use incredible amounts of fresh water and turn human waste from a material resource into a financial and human health liability. We need nutrient and energy recovery systems that make the best possible use of our natural resources and improve our quality of life. Rather than trying to keep patching up a broken sewer system, we need an entirely different approach to human waste.
In response to the growing understanding that taking responsibility for waste produced by humans is essential to sustaining us as a population, our project Jester works to challenge the individual to consider the implications of their personal waste and repurpose it to generate energy.
Our team decided to focus on generating energy from human and household waste when we began doing research into anaerobic digesters. These machines use microbes to decompose organic matter without oxygen, which creates biogas. Biogas can be used just like methane for burning in energy generation. We met with a team of researchers at the university who are working on a new kind of biodigester that harnesses the digestive power of ruminants like cows. This is an image of the lab model biodigester that these researchers are currently using. Nutrients like human waste and food scraps are fed into a tank which contains ruminant microbes taken from a cow, and those microbes then break down the difficult to digest cellulose, and that material is then passed into an anaerobic biogas generation container. This lab model digester can convert 75 liters of methane from 1 liter of organic waste, which at this small scale is the lower end of this digester's efficiency and potential performance. When this technology is scaled up and used in municipal waste streams, it could generate 40% more power at a 20% reduced cost compared to existing anaerobic biodigester systems.
The container was designed to blur the boundary of our relationship with human waste. It is created to be beautiful and feel just like any other furniture we created 2 versions of the container, with one being the larger one that's catered toward larger households and the smaller size for an individual or a couple. It is made to fit at the side of the toilet without taking up a bunch of space. This also allows for easy access and removal of the container. There are not much technicality in the container itself. We are using the existing technologies of a vacuum toilet that sucks the waste into the container while using minimal water. The smell and excess air can be sucked out and into the existing sewage system. The contents can be viewed from the outside. We are not trying to make the poop go out of sight, out of mind, but instead allow people to see where the waste is going, and they can also know when it is full and needs to be taken out.
How it works. First, solid human-generated nutrients, poop and organic household waste, are collected in the home. When you use your Jester vacuum toilet, your waste is diverted to a holding container. Second, the full nutrient containers are put on the curb for a weekly pickup. Local community members who help run the Jester Bioenergy service will stop at your house and bring your containers directly to the biodigester. Third, nutrients are fed into the biodigesters, generating energy and liquid fertilizer. Through anaerobic digestion, the organic nutrients that you produce are broken down into methane and other byproducts. Fourth, customers are materially compensated for their nutrient contributions. They will receive offsets on their energy usage based on the amount of energy their waste produced. Vacuum toilets, commonly used on airplanes but not often found in homes, are flush toilets that use suction for the removal of waste, both liquid and solid, resulting in minimal requirement of water. Not only does it reduce your water bill and reduce water consumption in your community as a whole, it makes the transportation of waste in the Jester container much easier because it limits the overall weight.
Here is how much energy we could generate with our proposed model. The average person in the US generates 2.57 liters of poop and 1.5 liters of food scraps per day. Using the conversion figures from the ruminant biodigester, this would generate 232.5 liters of biogas per day, which would create 0.465 kilowatts of electricity per day. Less than half a kilowatt may not sound like much, but it can actually count for about 15% of your daily electricity use. This is my energy bill for my 2-bedroom apartment that I share with 2 other people. We could generate a combined 3.6 kilowatts of electricity per day with all 3 of us. That's a significant potential savings currently being flushed down the toilet and thrown out on the curb. The savings from this human-generated energy could be passed back to the consumer in the form of a discount on your electricity bill, for example. Now, instead of your waste being something you have to pay to be rid of, you're materially compensated for generating value from your own body and your daily life. The biogas energy generation system also cuts into externalized costs that we don't always see connected to our utility bills. Because our system uses vacuum toilets, your water use would decrease by about 20%. After the energy is harvested from the digestate, you can treat the human waste to kill off harmful pathogens, as we currently do, and then sell the safe digestate as compost. Sewer system replacements and upgrades to deal with contamination are increasingly costly and likely only to get worse as our ecosystem destabilizes, and our system completely removes the most hazardous element of the sewer system, pathogens in the poop. Why keep trying to fix the same broken system?
As for concerns, such as waste leakage, the biodigester system is no more or less safe than current practices. It is not the waste that is changing, it is the process and what we are doing with it. While our current system focuses on waste disposal, or disappearism, so to speak, the Jester system decides that our waste should no longer be wasted. Though it is a small contribution, a part of our larger contemporary effort to shift to more sustainable ways of life, every little bit counts. The new system will not erase the current or lingering issues inherent of the old system, but it can ensure that they do not happen again. Through our research, we found that the current attitude surrounding waste today is largely one part association and one part assumption. Waste is typically associated with disease and the general idea of grossness. The assumption of our current system is that we are basically being protected from our waste and that is being taken care of. We have little agency over it and little interest in it, and thus the understanding of it is, essentially, that it is there and then it is not. Most importantly, this system, despite a lack of knowledge into its processes, is accepted simply because it is the current standard. Therefore, as with other new and unfamiliar products, such as smartphones once were, the right marketing and design can facilitate a transition to a new standard, the biodigester. Hence, our design incorporates a mixture of visual familiarity and the allure of a new and innovative product. In regards to the use of human waste for compost, while there is ongoing debate surrounding its adoption as a standard, the practice is already employed in impoverished areas of the world with little access to standard fertilizer. Current systems exist to process human waste to filter potential concerns such as hard metals and pathogens in order to turn it into good compost. Human waste is nutrient-rich, containing nitrogen and phosphorus, a higher concentration of which are in the undigested biomass byproducts of biodigesters. This biomass can thus be used for fertilizer, sold to farmers or returned to homeowners for use in their gardens, ultimately contributing to a circular economy. Regardless of its benefits, we recognize that there will be ramifications to this new system. We have largely pondered the cultural implications the new system may have. How may this new system change our perception of waste as being gross or diseased? Could the system cause people to imply that waste is suddenly more safe to handle? How might the treatment of waste as a value impact the economy? How will that perception change how waste is discussed? As in to say, if waste is valuable, will it be a private matter? To what extent can it be a private matter? How could it alter the way those, such as heavier or taller people, who produce more waste, are perceived?
Michigan judges, if you have questions, please go ahead and turn on your videos. I really enjoyed your video, by the way. Thank you. Thanks for presenting it.
Thank you.
We'll start with Leslie and follow up with Ali. Please go ahead, Leslie.
Hi. Hello from Ann Arbor. I'm there right now. Team, so U of M, go U of M. Anyway, how had you thought about how your biogas would be delivered to the utility company? So
yeah, so essentially, like the way it would be, the way the service works is that sort of the waste is collected in the home, but then it is transported to a waste management facility. So it's already there, essentially, if that kind of makes sense, I guess. Like the, yeah, like the actual at home part is more, yeah, like just kind of waste collection, and then it's sort of transported elsewhere.
So that way it just kind of removes the issue of kind of like wide scale issues that are inherent of the current system, like for example, like blockage and people putting things that they're not supposed to down the drain. And yeah, just the overall, like what was input in the video of taking responsibility for your own waste and taking.
So when they pick it up, you said it's picked up and that's when it would get delivered to the biogas would get delivered.
Yes.
No. Okay. By the way, I thought all the considerations you discussed, the psychological considerations and some of the others were really, really terrific. I want to give you a hand for that one.
Thank you. Yeah, thanks. We were really about the kind of, a large part of our project was about the more sort of personal aspect of it, like the cultural aspect of it, because like the scientific part is existing, but the current way in which it's being kind of put into use is either kind of working with the current system or still just kind of rough around the edges and lacking a design perspective. And so isn't as, I would say, appealing to the larger public. It's more, on the individual scale, something that people who are just super passionate about sustainability might do. But yeah, that's sort of like what our project was more based off of, was trying to push it into, exploring how it could be pushed into the larger sphere, those who don't really know anything about waste management, are not really concerned, that sort of thing.
Well, the way the toilet looked, it would be helpful because it's real familiar. Thank you.
Exactly. Yeah. Thank you.
Thanks, Leslie. We'll now move on to Ali. Please go ahead.
Yeah, well, if all else fails, I think you have a great career for doing voiceovers. It was really great. It was a really great video, and it was very clear what you were proposing. I'm not sure why poop is such a taboo topic, given it's something that we all do every day. It confuses me why it's such a taboo thing, when actually we could learn so much about ourselves, our own health, the health of our communities, if we could actually, gauge some of that data and use it for positive purposes. So I think my question is a little bit about, you've made this a very kind of individualized system, and that it sits next to the toilet that you've just pooped in. But I'm wondering if there's a way to kind of utilize existing infrastructure to actually, do what you're proposing, but without actually redesigning the entire system. And then in line with what I just said in the beginning, that we can learn so much about our poop. If it is individualized, is there a way that we could somehow visualize that data from our own, gut biomes, to understand how we can improve our health or the health of our family?
Yeah. Okay. So for the first part of your question, sorry, because you run it back with me again. Sorry. Maybe it actually might be easier to go backwards, perhaps. So in terms of sort of seeing that information, we consider that for, like, there came to be a point in our project, since we were kind of focusing more on the cultural aspect, and so obviously there's a lot to consider. There was a point in our project where we had to decide, okay, let's slow down, let's not go overboard with this. But we had considered this would be a really great product to, figure out a way to have people be able to self-monitor even just, the health of what they're eating and things like that. But that even just in itself opened up an entire other, which can be addressed, entire other bag of questions of okay, how can we mitigate, because this whole project, a large part that be a concern to people is privacy. Now, technically speaking, waste is already not a private matter. I know they already used waste to sort of monitor COVID in the local area, and I know at least in Ann Arbor. But once you get down to, not having it all mixed together and it being much more individual, it becomes a lot more personal, a lot more privacy concerns for people. But we had considered okay, this would be a great way of making sure that people are getting, for someone to be able to self-monitor how many vitamins they need, making maybe doctor's appointments a little bit easier, a little more straightforward. So those were considerations we had made, but we were mostly sort of focusing on like the basics first before we moved on to, further alterations. And then what was the first question? Sorry.
The first question was about utilizing existing infrastructure.
Yes. So part of
there's a big thunderstorm here. I don't know.
Wow.
I'm gonna mute myself.
It's okay. Yeah. So part of the ramifications, we didn't put it in the video, but like one of the ramifications we thought of is, in existing infrastructure, like what happens to all those pipes, things like that is eventually essentially becomes litter. And kind of like what we had slightly touched on in the video was we kind of came to the conclusion that, some of these things, like the existing system, it has its weaknesses, as we discussed, and has to be cycled out eventually over time. And so it wasn't like we hadn't totally figured out like a complete solution for it, but we realized that is a solution that has to happen regardless. And it would be better to have that solution, happen once rather than every single time or every couple of years like it does now. That's what we touched on sort of towards the end, is like we can't, some of these things have to happen regardless. Like we touched on, like Detroit has to redo some things and fix a lot of things, but the kind of essence of our project was at least it doesn't have to happen again. But yeah, I mean, that is part of like what we were considering. But again, like I said about the sort of hitting a point where we had to kind of slow down a little bit, we started thinking like, okay, what could we do with those pipes? Like what could be, like what can we transform those into? But then that was like a whole other project. So yeah.
Yeah, thank you so much. I'm all for generating passion around poop. Great project.
Thanks for your question, Ali. Thank you for presenting, Jester. We're going to move on now. We're out of time. We're going to go to Savannah College of Art and Design. Thank you both.
Thank you so much, Biodesign Challenge.
Microplastics are microscopic pieces of plastic that are around 5 microns or less in all directions. While microplastics research is still relatively new, many researchers and organizations are working hard to better understand how exposure to microplastics through air, water, and food is causing negative health effects in humans, animals, and the environment. So far, microplastics have been found to cause hormone disruption, sperm deformities, and low fertility rates in mammals. They are found in 83% of tap water worldwide, as well as in human blood and placenta, and their long-term effects are not fully understood. There are 2 ways microplastics are created. Primary microplastics are small plastic components or products that are intended to be small, such as beads, glitter, or cosmetic ingredients like microbeads or synthetic mica. Secondary microplastics are formed through plastic degradation and erosion. The main contributors to secondary microplastics are manufacturing processes, plastic breaking down in landfills and aquatic environments, and friction, such as washing synthetic clothes or shoes and tires rubbing against the road. In our research, we conducted over 25 interviews with consumers, researchers, manufacturers, and government representatives. We surveyed over 50 individuals and also conducted a public, interaction-based focus group. From the data that all these primary research tools generated, we were able to compile over 2,400 data points, which we then affinitize into our final framework. The framework is broken down into 4 major categories: sensory prompts, benefits, features, and core emotions. Sensory prompts refers to that which people generally associate with microplastics. We found that first, people understand that microplastics are closely linked with synthetic textiles and apparel. Second, they think that there is a lack of transparency around plastics in general. Third, they associate plastics with not knowing whether their attempts to make good choices are working. And finally, people know that microplastics don't go away, they only get smaller. When it comes to the benefits people desire, we found that people want to be able to make better choices as consumers, know the impact of their efforts, and ensure that they are healthy and safe. The heart of our microplastic study can be found in the core emotion section of the framework. This section describes what people deeply want. Through our research, we found 4 core emotions. First, people want to feel optimistic that plastic use will decrease. Second, people want to be sure that there are no microplastics in their drinking water so they can feel secure about their health. Third, they want to be knowledgeable about the environment, the problem of microplastics, and the current innovations surrounding it. Finally, people want clear, concise, and accessible information to improve their trust in governments and businesses. We realize that the features necessary to meet these emotional goals are solutions that should be educational, collaborative, motivational, and transparent. Our solution, Sunbox, is an innovation directly derived from our framework. Sunbox is a countertop appliance that uses UV radiation to chemically break down small or thin plastics, such as single-use plastics, into carbon and trace amounts of carbon dioxide. By learning to use Sunbox, people will become more knowledgeable about microplastics as well as plastics in general. They will gain the ability to control where some of their plastic ends up without having to rely on processes or institutions they don't trust or understand. While we can't stop plastic from being produced yet, we can stop it from ending up where it should never be, inside our ecosystems and our bodies.
Welcome, Kira and Olivia. Thank you for a really interesting video. Judges, if you have questions, please turn on your videos. Sukanya, please.
Hi, team. Great presentation. Yeah, plastic recycling is a joke. Let me just start there. So my question is, using your Sunbox, how much of plastic can you break down and how quickly?
Yeah, so through our research, we haven't created the thing, we haven't done any testing or any kind of stuff. So most of it's just been done through academic research. So what we found is that mostly polyethylene and PLA break down pretty fast, but that's just where the research has landed. So because PLA is technically a bioplastic made from starch, it'll definitely break down faster than polystyrene. But so what we've found is our device is more for like food waste. So that's why it's more of a countertop device. So like I always think about the plastic that comes on American cheese or like your grocery bags or that thing of salsa that you dug into from the deli, like those kind of plastics. So really thin, easily breakable, but digestible plastics that wouldn't see the sun if they were in the landfill.
Thank you.
Carolina?
Hi, thank you for your presentation. It's interesting. I've been looking at the compost like home machine. I don't know how do you call it from Vitamix, and so you can put it on your counter and it composts organic waste. So I was thinking that it could be a good fit with that. So how does it work exactly? Because I wanted to know a little bit more of how you envision this to work and how would you make it a scalable product?
Yeah, so we're not totally sure. So there's like 2 different ways where the plastic would degrade. So we're trying to kind of mimic ocean degradation. Because that's where a lot of microplastics are made that we learned through our research. And we were like, well, how does that happen? How does it break down in the ocean faster than really anywhere else? And we realized that was through the UV radiation. So sometime in the aquatic environments, the carbon is like made into water soluble carbon. So I'm not sure if we would need to add water to the machine or if it could just be the radiation with like a fan going. Because research has found that the temperature difference does not necessarily change the plastic degradation. It only changes it by about 25%. So keeping the temperature regulated and trying to regulate the environment in which it is in could accelerate the plastic degradation.
Thank you.
Yeah.
Great. Judges, I can ask a question. Can you talk a little bit about that survey you did and all those Post-it notes you collected? What were the 1 or 2 key takeaways from that experience?
Yeah, I'm gonna hop that over to Kira. She was actually in the video showing you our sensory cue, so she can chat about that.
Hey, yeah. So people, I think our biggest takeaways were that people don't really feel that they have a sense of control over their plastic consumption or their plastic disposal. That was overwhelmingly the message we got, was that, people would talk about different mitigation strategies that they had, but they would always kind of come back to like, I don't really know if it's doing anything. I don't know if it's helping. I don't know what else I can do. I don't know what I should be doing. And that's a large part of what gave way to the idea of Sunbox, was giving people something that they would have control, and at least for like a bit of their household plastics, be able to deal with it themselves, understand how it works, and know where it's going, and say like, okay, at least this much plastic is not going in a landfill, is not turning into microplastics, is not going into a recycling system that's very opaque. So I think overwhelmingly that was our biggest takeaway, was that like, people don't feel like they have control, and they don't feel like they have the knowledge ability to make those good choices, or they don't feel they have the knowledge ability to figure out what they should do next, really, even if they really care about the issue.
Great. Thank you both for your project and for joining us today. We're going to move on to our last team of this session. This is Ball State. Let's roll it. Thank you both.
Right now, it feels like our designs are short-sighted, created only for this passing moment. They are easily discarded for the next flash of aluminum, plastic, copper, or silicon. What would happen if we expanded our perspective? What if we focus on something bigger, something more sustainable than just short-term gains? What if we thought of design as a way to create better communities that could heal divisions between people, better communities that could support different species, and better communities for a more sustainable environment? If we wanted to design with all communities in mind, where would we focus our attention? Rivers, lakes, streams. They give us life. They are the foundations of our communities. However, according to the United States Geological Survey, more than half of the nation's streams are in poor conditions. Current agricultural practices and urban runoff are key stressors to our river systems. Over 40% of the nation's river and streams have high levels of phosphorus and nitrogen, which can cause toxic algal blooms. Excess levels of nitrogen and phosphorus cause acidification, reduce oxygen levels, and create poor conditions for all life. One example of a river with a problematic history is the White River in Indiana. The White River begins as a stream in Randolph County, flows from Muncie to Anderson, and continues to the Mississippi, where it eventually pools into the Gulf of Mexico and the Atlantic Ocean. Unfortunately, the condition of the White River has been unstable for over 100 years. Settlement occurred along the White River in 1886. Since then, the discharge of pollution has been a major problem for the environment and the local community. On August 14, 1916, the city of Indianapolis and 8 businesses were sued by Morgan County farmers for fouling the White River. They accused the city and packing houses of dumping refuse directly into the river. According to the Indianapolis News, the conditions of the stream had become so filthy in the summertime that people along the river could hardly remain in their homes. The livestock would not drink the water, and the fish died in great numbers. It wasn't until the Clean Water Act of 1977 that the White River saw significant improvement. The federal regulation helped establish testing and enforcement agencies which addressed some of the issues caused by raw sewage discharges and agricultural runoff. However, on December 12, 1999, the Herald Bulletin reported a chemical discharge at the Anderson water treatment plant. 1.6 million gallons of toxic water from a headlight manufacturing plant were released into the White River, which caused the death of over 4 million fish and polluted the river for a generation. Ultimately, the company was forced to pay $14 million in a legal settlement, and $6 million were used to help clean up the White River. Today, the White River still has many problems. It continues to gather pollution from fertilizers, herbicides, and pesticides. Dams block aquatic life activity and limit ecosystem overlap between parts of the river. Phosphorus and nitrogen are still problems, and heavy metals still permeate the water. And throughout the river, overflows containing raw sewage as well as stormwater runoff from streets degrade the water quality. Considering that the last 100 years of development have been so detrimental to the White River and its communities, it's important for us to reconsider our relationship to the natural world. How can we use design to build better communities for the next 7 generations? Introducing the Hypergarden. The Hypergarden is a speculative design project that seeks to improve the White River in Muncie, Indiana. It is a floating garden built with bamboo, cattails, sunflowers, and lavender. These plants are capable of removing pollutants through their roots, which is a process known as hyperaccumulation and phytoremediation. As a garden, this project helps promote a stronger ecological relationship with the community and the White River. As a gathering place for humans, animals, and plants, it is the foundation for a stronger community. Phytoremediation is the process of using living plants to clean the soil, air, or water contaminated with hazardous materials. By harnessing the natural capabilities of plants, contaminants can be removed, degraded, or stabilized. It can be a low-cost but time-intensive alternative to traditional remediation on-sites where toxins are present at shallow depths between 6 to 12 inches. At the Hypergarden, sunflower and lavender are used to remove heavy metals from contaminated soils. The common sunflower can hyperaccumulate nickel, copper, arsenic, lead, and cadmium. Lavender is capable of remediating lead, cadmium, and zinc. By including these flowers at the Hypergarden, we hope to educate and inform the community about the ecological issues at the White River. The main extractor is the cattail because it is effective at hyperaccumulating phosphorus, nitrogen, and other heavy metals. Researchers have found that the cattail is capable of removing 48 to 99% of zinc, lead, cadmium, iron, and nickel at constructed wetlands. For some, the use of cattail may sound odd or counterintuitive. In many parts of the US, it is considered an invasive species. One of the primary reasons for this is because cattail release high seed counts and can spread quickly in areas where phosphorus and nitrogen are abundant. In these areas, we may need to rethink our existing notions about cattail. We might need to ask why cattail is growing so rapidly. If we reconsider cattail as a telltale sign of excess phosphorus and nitrogen, then its presence would be a bioindicator of pollution problems. One of the most convincing success stories of cattail was produced in the 1990s when the state of Florida began using it to prevent excess phosphorus from entering the Everglades. Specialized bioremediating wetlands called storm treatment areas were created to accumulate nutrient-rich stormwater runoff. Today, over 56,000 acres of land have been converted to these specialized wetlands that are abundant with cattail. In 1 year, it is estimated that 100 metric tons of phosphorus could accumulate in these storm treatment areas. And in the 23 years since the beginning of this project, these wetlands have prevented 2,300 metric tons of phosphorus from entering the Everglades. In other parts of the country, researchers are currently reconsidering the role of cattail in the fight against climate change. At the Salk Institute, researchers are studying how cattail can act as a carbon sink by removing CO2 from the atmosphere and storing it in wetlands. If we reconsider our preconceptions about certain species, we may be able to implement new systems that can benefit our communities and repair our environments. While the Hypergarden proposes using cattail as a nature-based technology, we know that planting a floating platform won't solve all the issues at the White River. That's why it's important to consider that the Hypergarden is a place to gather, to share, and to rebuild. Hyperaccumulation is also a metaphor for community. Through public activities, it could be a new space for us to meet, learn, play, observe, encounter, laugh, love, cry, find sustenance, and to think about the future. We hope that the Hypergarden becomes a space to accumulate knowledge and reconsider what design could be, if it was focused on what we need for the next 100 years. The Hypergarden is a speculative design project created by students at Ball State University that encourages designers to reconsider what technology looks like and how it will impact the future. According to Liam Young, we live in a world of competing narratives and stories. He says that we need better stories about the future, stories that are focused on renewal and change.
In many ways, fiction has always been an extraordinary shared language. You know, we've always, since we can sit up, stared at the TV, watching films or TV shows. We've fallen asleep in the pages of a novel. And it's really the way that our culture shares and disseminates ideas. So I think in our work, what we do is co-opt the mediums of fiction. We embed important ideas about the futures that we're all going to be living in together. And I think that if we can be clever about using fiction and film as a new kind of site, then we can design and prototype a whole range of possibilities about what the future might be, and put them into the world with enough force that they might find traction, or that at least they might connect with audiences so that we can have a shared conversation around the futures that we all want to be living in together.
Through the act of speculative design and world building, the Hyper Garden imagines a future where bioremediation is a natural part of our built environment. This type of design asks us to reconsider our current preconceptions about species like cattail and seriously think about how they might help us adapt to our changing environment. According to Julia Watson, it is clear that our connection to nature is broken. The environmental threats that we face have reframed what we as designers should be working towards. To do this, Watson proposes incorporating traditional knowledges and nature-based technologies into existing design practices.
Technology can be different. Technology can just use nature and the systems that nature is composed of to do things for us, like clean sewage, to provide drinking water. That's architecture that is beautiful and emblematic of a particular place. We can be informed of how to look better through the way that we build our environments, whether they're urban or rural.
If we take seriously the idea that our greatest responsibility is to be good ancestors, then we need to rethink what biodesign could be. What if biodesign is the substrate for more vibrant communities? What if it was part of a larger system of ideas that refocuses our relationship to technology, to each other, and to the environment?
Welcome, Claire and Zeke from Ball State. Great video. Judges, I'm sure you have lots of questions. Please turn on your videos. But I guess I'll just go. Carolina, you can follow. I'm interested in why you chose a sort of a floating garden as your solution. How did that become your speculative solution among the many options you had available to you?
We mainly found inspiration in the floating gardens project that we had from a classmate that we had, and they used low-tech design to use these growing structures from willow trees and steel. Over time, the structure would grow, and it was like a really interesting combination of use of architecture and nature, and something we wanted to implement as well with our project through natural resources.
Yeah, not only that, but we have some design precedents like the Living Roots Bridge, and that was kind of discussed in the video by Julia Watson, how it kind of integrates just nature and kind of like that community aspect.
Great. I'll pass the baton to Carolina. Please go ahead.
Thank you for your presentation. Yeah, I think that with what you touched on your presentation in regards to storytelling, I think that was a powerful narrative. So could you talk a little bit more about how storytelling is compelling in regards to your process and your design at the end? What was it that really marked the narrative and you brought it to that speculative space?
Yeah. So I guess the aim of this project was just to kind of bring the local community together, reframe the narrative of cattail on a case-by-case basis, and kind of raise awareness around the White River. And so through this like storytelling aspect, we wanted to really integrate and I guess create empathy so that we can just have a greater appreciation for life and just sustainability.
Thank you.
So,
oh, sorry, I cut you out.
No, it's okay. I was going to elaborate a little bit more. I was going to say the hybrid garden is just as much about engaging the community as it is about like the architecture, so like the actual structure itself. We not only want to create a story, but like build an environment where people can relax, reflect, and reconnect with the world around them.
Thank you.
Thanks, Carolina. Ali, please go ahead.
Hello, thank you for sharing your project. I'm questioning why you're posing this as a speculative design project when it seems this is something which could be integrated now.
Yeah, for the speculative design, we kind of thought more conceptually with the idea of concerning with future design proposals and critical nature and how this can affect the future as well. So maybe this could be implemented now, but we're not really sure about the whole time frame. We just really wanted to be really conceptual with the whole process and start there.
Yeah, and also like we didn't have the actual materials and resources to kind of go that route and see if it's something we could do now. So we've kind of took the speculative approach for this project.
Yeah, and I understand that you're pushing the community side of the project as well, which I think is fantastic. But on a more practical level, have you thought about the fact that gardens tend to be seasonal? And I don't know anything about this one particular plant that you're talking about, but especially with sunflowers and lavender and these kinds of plants, they're not necessarily there all year round. So there was that. And then the second part was about where to place these gardens on the river. So is there any evidence as to where these gardens would be more effective? You know, if you were to plant a network of these gardens, for example, I don't know if they would be more effective on the bend of a river or where the water flows quickly or slowly. Have you done any research into that?
We've researched it, but we haven't really thought logistically about where these locations, these beds would be placed, but we know we would want to like have them networked around the White River. Not necessarily where, but we also know that sunflowers and lavender can be difficult in certain environments, but they're mainly there to also, as they're an attraction point to get people to be engaged with the community as well, since many people know what sunflower and lavender are as well. So they're also there to fight remediation, but also to get people to come to the hybrid garden.
Yeah, in addition to that, like the cattail is like a good bio indicator of areas with high polluted metals like lead, cadmium, zinc, phosphorus, nitrogen, and other heavy metals. So the cattail will kind of just help us assist in where like what places would actually need this platform to be placed on. And so although like gardens can be seasonal, using the dried cattail could be another way to kind of help use that same material and kind of recycle it to continue building that structure and just maintaining the structure in general.
Great. Thank you very much, Claire and Zeke. We're out of time. Thank you, Ali, for your question. We're now going to move on to a 15-minute break. Vina will be taking the role of MC when we get back. Excited for that, Vina. Thank you all for spending these last couple of hours with us.
Hi, I'm Leslie Ruckman. Hi, I'm Gal Nissim. I'm an artist and a scientist. Birds come to visit the city while they migrate, and they don't see glass and they collide into glass.
Through the animations, we're exploring this idea of the collision with the glass. So you'll see birds flying, hitting the window, and this is because they don't see glass. It's a concept that makes sense to us. It doesn't exist in nature. And zooming out from that, on the other glass windows, these UV murals that are invisible to our eyes, you're bringing to life what birds can see with their own eyes because they do see the UV spectrum. So these murals make the window visible to birds and hopefully helps them avoid collisions in the future.
The action of Rosa Parks, the words and leadership of Martin Luther King Jr. inspired me to find a way to get in the way. And I got in trouble. Good trouble. Necessary trouble.
In 2012, a 17-year-old Trayvon Martin was visiting his father in Sanford, Florida. During a short walk from the local store, Martin was stalked and gunned down by an ex-neighborhood watch captain, who was eventually tried and acquitted. This trial and result created a ripple of sadness and disappointment through the Black community. The Black community responded with the hashtag #IamTrayvonMartin, a social media protest campaign that featured celebrities and allies wearing hoodies, the outfit that was believed to have caused the profiling of this black boy walking through his neighborhood. Shortly after the acquittal of Martin's murderer, the phrase #BlackLivesMatter emerged on Twitter. This movement gained momentum as a social justice juggernaut that sought to center the disparity of threat to black lives in our society. The hoodie became a symbol of every person of color who had experienced racial profiling while simply existing. Subversive, biofashion for Black lives. We began our project as part of an Afrofuturist speculative design process that concluded with inspiration from the Biodesign Challenge, Say Goodbye to the Wind by J.G. Ballard. We were challenged to find or create representations of ourselves in this dystopian future. After further exploration of many variations of biodesigns to support Black lives, we landed on our current project presentation. Our group was divided into 3 teams: materials, sensors and schema, and design, each supported by a Spelman faculty member within the discipline. This project features a speculative and critical wearable design that will integrate wearable sensors that can collect biometric data from the wearer. The individual data collected can be crowdsourced across urban landscapes and used by entities designed to provide therapeutic and protective response mechanisms. This biodesigned garment is meant to be a subversion of the hoodie, typically worn throughout the Black community. Through our speculative upcycling of the typical hoodie, we envision an Afrofuturist wearable design to change the narrative of the social impact of wearing a hoodie while being Black. We began our process using a variety of materials and processes in our iteration to find best examples of the ideas we wanted to express in this garment. Limitations of our isolated existence has made it difficult to explore many physical prototypes. Thinking about the material for our hoodie, we first explored the characteristics that we wanted our hoodie to possess. The first was physical protection. This desire led us to explore materials that resist force and pressure. Accordingly, the outer material is modular and comprised of a matter made of spider silk, silk leather from Tufts University's Silk Lab. This layer is rip, puncture, and water resistant. However, the fabric for our hoodie is triple layered. The second layer, encased in the outer layer, is made of a compressed mixture of starch and water, oobleck, to create a polymer with non-Newtonian properties to encase the flexible electric components and provide the wearer with protection from blunt force trauma without sacrificing mobility. Finally, the inner layer is made of a removable conductive mesh that would connect the different modules together as a network. This part of the garment could be removed and can easily be washed. The various sensors within our hoodie measures a range of biometric data, including body temperature, heart rate, blood pressure, breathing rate, and it also provides an EEG of the wearer's brain when the hood of the garment is pulled up and over the wearer's head. This is all in an attempt to determine the wearer's state of well-being. The data obtained by the hoodie sensors with regard to body temperature, heart rate, breathing rate, and blood pressure will then be used to reflect the wearer's state of well-being in the arm and hand band of the hoodie, such that a calm state is represented by blue, a nervous or slightly irritated state is represented by green, a fearful state is represented by orange, and panic attacks or cases of extreme fear are represented by red. The color-changing aspect of the hoodie is premised on an if-then-else loop, such that if certain biometric data, as measured by the sensors, falls within a specific range as stated in the code for the loop, the color of the hoodie's hand and arm bands change, using LED motors as stated within the code, reflecting the output for that range.
We envision our design existing as an embodiment of historically rooted community connectivity, in the same way that the grapevine has been used to share community networks around sentiment, threat, and well-being. This hoodie would allow for members of the community to share and propagate data that would allow for support networks to engage as needed. Additionally, wearers could be community members who are involved in various forms of engagement or activism, whether primary actors who are witnessing events of note or those who are secondarily engaged. The data can be encrypted and securely shared and sourced across landscape and utilized by local community organizers, leaders, and support structures who are providing care and support. According to the UN, conflict, insecurity, weak institutions, and limited access to justice remain a great threat to sustainable development. By respecting human rights in this time of crisis, we will build more effective and inclusive solutions for the emergency of today and the recovery for tomorrow. We believe that our subversive biodesign will afford Black communities the opportunity to create a more positive well-being among its members, specifically those facing threats of brutality and marginalization.
We, the Spellman Biodesign team, would like to thank you for acceptance of our biowearable designed submission that seeks to support peace, justice, and inclusive societies as part of our commitment to Representative John Lewis's charge to find and embody necessary trouble. We found this work to be incredibly thought-provoking, as it challenged us to see ourselves in a society that promotes the well-being of Black lives. We'd like to thank our advisors and teachers for their support, and we thank you for your time.
Back, everyone, to session 2 of day 2 of BDC Summit 2022. That's a lot of twos. My name is Veena Vijaykumar. I'm BDC's program director. I'm going to take on from Dan, who is doing all the MC this morning. Welcome to this session. We're excited to see what the students have to come up with or have come up with. We're going to continue with the presentations in a second, but I wanted to send a few reminders first. Firstly, if you post about the summit, we do ask on social media, on Twitter or Instagram, we do ask that you tag us at Biodesign and use this hashtags BDC 2022 and hashtag Grow the Future. And we hope you still continue to vote for your favorite project for the Community Choice Prize on the BDC website. So going into our presentations, the next two teams that you will see are the finalists for another sponsored prize called the Science Sandbox Prize for Public Engagement. This prize encourages students to create projects that amplify public discussion about the multitude of impacts biotechnology can have on society. So we want to say thank you so much to the Science Sandbox, an initiative of the Simons Foundation, for your continued support of the students. We'll have Program Director John Tracy with us today as a judge, and we're excited to see what the students have to present. The two finalists for this prize are the Parsons School of Design and Robeson High School Owl of Future. And firsthand. We're about to see their videos right now, so let's start with Parsons School of Design.
Welcome to the Order of the Biodivine.
I'm Jess.
I'm Rachel.
And I'm Oscar from the Parsons School of Design. The Order of the Biodivine is a speculative, immersive, theater, and educational experience that explores the intersections of religion, science, and interspecies symbioses in an increasingly climate-insecure world. Set in a coming-of-age ritual in 2070, this project uses performance, mycelium artifacts, and a choose-your-own-adventure-style structure to encourage a new perspective on our relationship with biodiversity, genetic modification, and the natural world.
We envision this project as a 30-minute immersive theater experience for audiences over 12, followed by an educational exhibit. The event is designed to travel. It will be ticketed, and proceeds will go to local community science education. Today, we are presenting a theatrical trailer for the project.
Humanity, humility, clarity, humanity, humility, clarity. This summer, you're invited to Mycelium Melodrama, a speculative spectacle, a soon-to-be nature cult classic. The year is 2070. Life as we know it is gone. After years of political division, warfare, and natural disasters, the global north has ceased to exist. Simultaneous climate catastrophes in what is colloquially called the Big Shift have destroyed local and national infrastructure, governments, and economies. Billions of people, plants, and animals have died. But a few have made it through. Settling in what is now known as Old Rochester, this group of Big Shift veterans have survived by banding together in a theocratic community that uses genetic engineering to meet their basic needs. They value biodiversity and prioritize living symbiotically as their way of answering for past human activity. They are a community of equals, but one stands above the rest. Meet Sophie Washington, genius biochemist, charismatic leader, and self-appointed oracle of the Order of the Biodivine.
You and I know that we are not alone here. We are never alone. We are just one of so many interconnected entities in the spaces we inhabit. One small part of a bigger network of thousands of beings, all living and changing and grasping for life the same way we are. The natural world is richer, more complex, stranger than I could ever imagine. The more I study these plants and soils and mycelium structures, the more I feel like I don't understand the nuances of their communities. The wider and more complex our network seems to be. But I do know this: we are no more important than the earthworm in the soil or the pigeon in the sky. We need to take everyone's needs into account. We must live symbiotically.
She's a 90-year-old who's lived and lost, and she is determined to keep humans from making the same mistakes again. She's found a way to genetically engineer a new path forward, one she administers during an annual coming-of-age ritual for teens of a certain age.
As your divine oracle and leader of this sacred order, I now invite you to begin your humbling ceremony. Please rise. In this bowl is an ego remedy, a special genetic enhancement biogel that I created that allows us to sync with the mycelium networks. Once this gel is applied to your skin, you'll live in greater symbiosis with fungal networks around us. Some of them will start to live on you. You'll trade nutrients with them and electrochemically connect with them too. We use this biotechnology to plug our consciousness into our ecosystem, to haptically connect to root systems and get information about incoming climate events, infestations, and disease before those disasters hit our community. This biogel allows us to better understand how we can live together and atone for past mistakes. It will soon allow you to do the same.
Meet JM. This is his year to come of age. He's been preparing for this initiation his whole life, but now that he sees the ceremony up close, he's not so sure about Sophie's plans.
It's not the pain. I'm not worried about being in pain. I'm worried about being a hypocrite.
Humans don't easily lay down their bodies when other creatures might benefit from their suffering. They don't easily give up that seat at the top of the heap. They need to be humbled, reminded of their place as a part of nature and equal to others.
What makes you think these organisms want us around now? Did you ask the mycelium before you just plugged into it and started electrochemically interacting with it? You're playing God.
This is the only way forward. I do know that we have to do what we can to work with others to repair what we've damaged. And that starts with communicating with them so that we can better understand their needs. We have to take others' needs into account.
That's exactly what I plan to do. I'm going to sacrifice my body for the benefit of the fungi.
Looks like we're not the only ones who want to vote.
Join us for an immersive choose-your-own-adventure performance that will probe the relationship between humans and the natural environment. Sophie and JM will make a choice. So will you. Since we are working over Zoom today, we built out a digital experience that echoes the choose-your-own-adventure structure of the play. If you would like to choose your own ending today, please visit our website at orderofthebiodesign.com. You can also find more information about our project there. We designed the Order of the Biodesign to be transportable, easily set up in different environments like science museums, outdoor tents, and unused structures to reach audiences wherever they are. The sets will be created with a combination of production mapping and natural plant life to evoke a post-climate apocalypse atmosphere.
When they arrive at the performance, audiences will be invited to gather in an immersive space designed to appear as a laboratory turned ritual space, set in a post-climate apocalypse world. Over a 30-minute period, they will see our comedic, educational story unfold.
At the end of the play, as it is currently designed, audiences will choose to join Sophie's cult or die honorably with JM. They make their votes known by standing with JM or by remaining seated. Those who remain seated receive Sophie's biogel on their skin and experience the atmosphere around them changing. The sounds of mycelium communicating begin to surround them, symbolically representing their newly developed interconnectedness. Those that stand with JM move to a separate room where they see the future they were most interested in pursuing, an immersive video of plant life growing without human interference.
When the production ends, all audiences funnel into a third room with an educational exhibit that allows them to explore the themes investigated in the performance, including climate change, genetic engineering, and mycorrhizal networks, among others.
To ensure that the performance speaks accurately about the current scientific discoveries and their potential ramifications in 2070, we interviewed experts, read scientific articles and books about the topics, themes, and conflicts at the heart of our characters' world.
We also conducted a series of futuring exercises to explore potential outcomes and ensure our fictional world was rooted in today's realities. From there, we created a series of fictional timelines, narratives, and mycelium-based prototypes.
Though we are proud of our project so far, work still remains. We plan to revisit the script, writing in more characters and creating more opportunity for audience participation. Our current team is limited to specific backgrounds and perspectives, and we plan to expand our collaboration to include more diverse points of view, including exhibit and theater professionals.
Ultimately, we hope that we can use the comedy of the Order of the Biodesign to engage the public and expand educational opportunities around complex scientific topics. We've outlined a few of them here.
Thank you.
John, you're already here. I'm going to open it up to you for questions when you're ready. Please unmute.
Thanks so much, Nina. Thank you for having me. Thank you to the Parsons team. That was super interesting. I'll start with a sort of, I guess, basic question, maybe a two-part basic question is like one, what are some analogies like in the theater world that you were looking at when you were developing this? And I guess on a sort of basic level, like what do you want an audience member or participant to feel or experience when they go through the exhibit?
I can start, and then team, if you want to jump in. So I know that we were definitely looking at both sort of small scale regional theater productions, kind of guerrilla theater. I've spent a long time in New Orleans. There's a couple of smaller productions that are down here that take place in kind of alternative venues or with alternative structures. The other thing we looked at a lot were immersive spaces. So places like Meow Wolf in Santa Fe that kind of allow audiences to go in and explore different exhibits kind of as they'd like to without as much direction. And so taking a look at those kinds of experiences that we sort of personally had and other, that our colleagues have had was helpful in kind of inspiring what ultimately became kind of the form of this project. And I'm sorry, I should have written down the second portion of your question.
No, it's fine. It's really a second question. I was just curious, like, what do you want I mean, often in science engagement, like the kind of best projects, like you don't necessarily walk away with, thinking like, I just went through an educational experience. Like we often talk about like what we want people to feel when they're done with the experience. So what for you like is the ideal participant experience? Like what are you trying to get them to feel, I guess?
Well, again, I'll just be brief so that my team members can join in the conversation. But I think for us, when we started to explore these themes, we really thought a lot about how to kind of make them approachable, palatable, and engaging. Themes like bioengineering and genetic editing and the future of that practice, things like the relationship between humans and the natural world. And so this was sort of a way that we thought we could make those subjects a little bit more entertaining and engaging, and to have people kind of, I think at first, be sort of awestruck by the story and by the production itself, but sort of walk away thinking more critically about some of the themes that we're hoping to tackle.
Yeah, I think balancing the almost like Shakespearean aspect of the play, or like kind of that Greek tragedy style drama with an education space was something that was important to us.
Yeah, and I think that we definitely want to still have more comedy and even more interaction as well with the audience member. So in terms of what we want them to feel, I think we want this to be engaging and memorable and funny, to be honest, to be something they really enjoy going to and they learn the science kind of subconsciously along the way.
And sort of back to the previous question, some of the other precedents we looked at were the Sleep No More exhibitions. And then I was really interested in like immersive art also. So that's kind of where the projection mapping and some of those exhibition precedents came out of.
Yeah, that's a great reference, and I'm glad you mentioned the comedy aspirations. I did sense like this kind of tongue in cheek thing, so I'm glad you called to that. I'm curious too, like in the project description, you mentioned this play, this intersection between religion and biodesign. What about that kind of inspired you, or how did you make that connection? And yeah, maybe you could unpack that.
Yeah, so a lot of this project kind of was born out of some future speculation work that we did very early on in our process. And as we kind of began to build out our world and this sort of world that we sort of thought could be potentially happening in the next 50 years, we started to look at different types of institutions and what would happen on a political level, on an economic level, on an infrastructural level. And our conjecture was that in an age that's really marred by a lot of climate catastrophe, that a lot of the kinds of institutions that people rely on might go by the wayside. And so, this is a world that doesn't really have a government. It doesn't really have an economic infrastructure, a physical infrastructure for that matter. And so what are the kinds of things that people might coalesce around? And so I think that religion and this idea of kind of sort of a new old way of gathering became very interesting to us in this sort of the world that we were creating. So that was sort of one part of why we decided to move toward this sort of quasi-religious biodesign space. The other thing we thought a lot about was the character itself. Sophie, we wrote a whole backstory to her. We have a whole biography. And so I'd say part of it was informed by our sort of speculative thinking, and part of it was really character driven around kind of what this scientist ends up kind of becoming to her community. And it is sort of a quasi-religious figure.
Super fascinating. I know we don't have tons of time for questions, but Vina, do if you want to get
you have one more, if you have a burning one, you could do one more.
That's okay.
Okay.
Thank you all so much. That was super fascinating, and
Awesome.
yeah, thanks for sharing it.
Thank you, team.
Thank you so
we're
much.
gonna, yeah, of course. We're gonna move on to the next team, which is Robeson High School, Aula Future, and Firsthand.
This is our friend Jackson. Jackson is a happy, young person living in the United States of America. Say hi, Jackson. At first glance, Jackson might look like a typical person, right? But actually, he is a superorganism. We can call Jackson a superorganism because, like all the other humans on planet Earth, he is not made of only his own cells, but also many non-human cells too. Now, you might ask, what are these other non-Jackson cells? Well, the greatest source of non-human cells is actually found in Jackson microbiome. The microbiome is made of trillions of microbes like bacteria, fungi, and viruses that live inside and on Jackson's body. We are all full of them. But look closer. Closer. Where are all of his good bacteria?
Well, unfortunately, modern living patterns and behaviors have caused Jackson's microbiome to be way less diverse than it used to be. Important bacteria for his well-being are missing. Let's see. He lives in a city, so he doesn't often walk in nature. He doesn't have a very diverse diet, and probably even eats fast food too often for his own good. Also, since that pesky COVID-19 pandemic, Jackson has been using a lot of antimicrobial soaps everywhere. On his hands, on his body, way too much. And he has been using antibiotics too often, sometimes without consulting with his doctor.
Our modern day behaviors are taking a toll on our microbiome, so much so that many species of bacteria are slowly going extinct.
Microbacterium vagae used to be abundant in soil. Recent studies have examined its potential influence on the gut-brain axis, and they found that this microbe can reduce anxiety-related behaviors and increase serotonin. Without you, we will all be more anxious and depressed. Firmicutes carry many of our disease-protective foundational flora, such as lactobacillus. Beneficial firmicutes help with the fermentation of fiber, therefore helping digestion and nutrient intake. These beneficial firmicutes are almost missing too. Bacteroids ferment an enormous amount of complex plant fiber and carbohydrates. These bacteria had beneficial effect on our immune system and overall health. We hope to see you back soon on our skins.
Some bacteria are indisputably bad, but others boost our immunity, protect us from infections, and produce the enzymes we need to digest our food. Without these bacteria, we won't survive. We won't even be the same kind of humans that we see ourselves as today. Researchers now think that our microbiome is directly linked to everything: our metabolism, immune system, central nervous system, and even some of the cognitive functions inside our brains. The lack of a diverse human microbiota, especially in more hyper-sanitized countries like ours, may be the root cause of many problems affecting us today, like asthma, allergies, diabetes, anxiety, depression, and even some forms of cancer.
And Jackson's not alone in this. His behavior are all the same as many others. We surveyed a few of our peers and found out that there are even more people than we thought have behaviors that would negatively impact the microbiome. How can we make sure these cute and important microbes don't become extinct in or out of our bodies? Can we find a way to reintroduce these important microorganisms to repopulate our microbiome?
Introducing the Microbiome Repopulation Project. The Microbiome Repopulation Project is an initiative created by us, a group of 15 and 16-year-old students from Philadelphia. Our mission is to source, discover, and supply a variety of bacteria and microorganisms that improve our health and reintroduce them back into the microbiome of the people of our community. With this initiative, we plan to raise awareness about the important role that bacteria offer and strengthen the connection between people, food, nature, and microbes. The way that our initiative works is simple. 1. We collect a repository of near-extinct bacteria. We gather them from soils, food, water, and natural habitat of local and international origin. 2. We grow large quantities of these bacteria. This way we could study the effect of different strains of bacteria on humans and make them ready for human consumption. 3. We host our microbial cookouts event where we collaborate with local community gardens, farmers, and chefs to create our bacteria-infused recipes for community outreach and education. All our recipes use traditional food production techniques like fermentation, aging, and bacteria culture addition.
Why don't you come and check it out? You are invited to the Microbiome Repopulation Project's very own event, the Microbial Cookout. The Microbial Cookout is a monthly event where we share the yummiest bacteria-loaded recipes with our community in Philadelphia.
Let's take a tour of one of our favorite recipes. Today, we're offering a trio of probiotic full bruschetta as an entree. Fresh Bacillus mozzarella with Philadelphia-grown cherry tomatoes, our micro bruschetta with fermented mushrooms, and bifidobacterium-loaded blueberry goat cheese bruschetta. The different microorganisms of this entree blend together in a unique mix of probiotics for a healthy boost of mood and immune system.
Blue mac and cheese is the main course. Our soul food dish is full of fermentures for the fermentation of fiber into nutrients for the body.
The bacterial yogurt and fruit parfaits with local organic berries. This dessert is full of added species of actinobacteria and bacteroidetes that increase microbial diversity and fight inflammatory bowel disease.
And Proteo smoothies, the refreshing smoothie that keeps you away from allergies. There goes Jackson. How does it taste? Good, isn't it? Let's have a look at your microbiome to see if it's happy. Yes, look at them. They're so happy and cute. And you're making friends, Jackson? The community is really loving it. Everyone is learning about the importance of taking care of their microbiome and really enjoying good food. Everyone is really connecting with each other, nature, and themselves.
Learning about the possible extinction of some important bacteria in our microbiome and realizing how our lifestyle is endangering them has inspired us to take care of our health and to reconnect with nature and food. Even though the Microbiome Repopulation Project is still young, we have managed to raise awareness about the important role that bacteria play in our day-to-day life, and managed to strengthen the connection between people, food, nature, and microbes with our community events.
There is still a lot to learn about the microbiome, the importance of healthy bacteria, and how to properly use these microorganisms in different recipes, but this journey really changed us. And it changed Jackson as well. Jackson is happy, young, and healthy. He has reconnected to nature, his food, and to his little helpers that make him, well, him. Say bye, Jackson.
Job, team microbiome repopulation project. Let's start with John, and then judges. This team is actually going for the main prize as well. So please do turn on your mics if you all have questions as well. John, please start.
Thank you. Happy to start. Great video. It was super funny, I have to say, at the in memoriam for the bacteria section. I laughed out loud. I'm glad I was on mute. Super creative. I'm curious, where did the idea of a cookout come from? Why did you feel like it was important to do that? Yeah.
I'll take this one. So originally, this project started off as wanting to help people, and we kind of viewed it as, well, how do we help people and like get to know people about these different bacterias? So we thought about a couple different things, like a food truck and things like that. So the main idea was food. So we thought of, well, what's a good way to bring people together and like have food involved? And we thought about a cookout, because in a cookout, everyone is together, talking, laughing, getting to know each other. And we thought that'd be a good way to implement the goal of our project. So that's why we chose the cookout.
Yeah, I love that. And how did you, a cookout isn't necessarily a hard sell, but maybe eat these bacteria cookout might be harder. So how did you get partners, both in terms of, you said local chefs or whomever or vendors, but also people to participate? How did those partnerships work?
So it started out as we printed flyers and things like that, as a cookout, normal cookout. And then as people came and showed up, we kind of explained to them, what we were doing and what the whole goal of our project was, and explained to them, why these bacteria think are very good for you and how using these hand sanitizers and things like that are very bad, because you're killing these good bacteria, and kind of educate them and, just to show them, hey, it's good to use hand sanitizers, but just be wary and maybe use less of it because you are killing these very good bacteria that are helping you and your body. So, yeah.
And then one more question before you open it up, Bina. I'm curious, like when you were selecting the foods you wanted to use, right? Like what was that research process like? How did you kind of make those connections between foods that are readily available and maybe some microbiomes that we've lost?
I'll do this one, Jackson. So basically the connections between the food and the stuff the good bacteria that we put in the food. Recently, our mentor Marco actually showed us this video about blue cheese and how, since there was blue mac and cheese at the cookout, sorry about that. As I was saying, the blue cheese is a lot different from normal cheese because it keeps all of the good bacteria that's good for us inside of the cheese because it's aged and it takes a long time to make, and it still tastes really good.
That's awesome. Bina, I'll hand it over to you. I know there's this one's up for a couple of prizes. So I don't know if there's other questions, but thank you all. That was great.
Thanks, John. Let's go with Todd, and we'll finish up with Anya.
Hey everybody, really great project. I also really like the in memoriam section. So I'm in DC and like, Jackson in the video, I don't get out to nature as much as I'd like either. I was curious if you guys looked at, or if there are different microbiomes sort of living in the cities that impact us differently versus the ones that you're putting in the food.
Could you repeat your question, please?
Yeah, so I'm just curious that, because you mentioned that, we're not getting some of the microbiomes that you're interested in the food because we're not getting out sort of into nature, or some of them have gone extinct. So I was wondering if there are different microbiomes or microorganisms in cities where people live that, interact with us differently, if you guys looked at that at all.
Well, yes. So like in nature, and like a lot of these foods are from like local gardens, so we get the bacteria from those, and also like our microbiomes, when we touch people, interact with them, we gain the bacteria that are on their skin as well, or their bacteria on their skin, which are good for you, which kind of connect to our microbiome. So it's a microbiome is a mixture of mothers and people around us and just the environment that we interact with. So it's all different depending on where we go and things like that.
Onye, please go ahead.
Hey team. Yeah, great project. I think that I could see like a clear connection between like hand sanitizer use and, you know, maybe people like starting to eat with their hands more or something like that, eating your foods to repopulate, you know, their skin microbiome. But my question is more around food safety of like, you know, you are working with a food product. I'm curious, like what sort of protections or what sort of like approach do you have to make sure that what you're adding to the food is safe for consumption and it is what you think it is?
Jackson, let's answer this one together. I'll go first. So basically, how we distinguish what's good in the food and what's bad in the food is where we get it processed, or actually, no, not processed. We're actually where we get it, or if the types of bacteria that we put in the food. So naturally, we only want the good type of bacteria in our food. So we basically only search for all natural food that either has the good bacteria in it, or we just put the good bacteria in the food ourselves. Take it away, Jackson.
And also just to clarify, like, you know, you could have the intention of thinking like, oh, we're putting this one bacteria in, but is there, you know, there could be contamination in that batch since you're like growing it. So just wondering if there's any food safety kind of like protocols, or if you're like buying these things off the shelf and putting it directly in.
Well, when we were thinking of this project, we didn't really think of that aspect of it, but we did think of it as well, like briefly. We did think of it as like, I don't know if you travel a lot, but you know how if you go to a country, when you drink these waters and things, the bacteria in the water, it have a different reaction to you because you have to get used to them. So we thought of it as kind of like that, but we didn't really have a full in-depth discussion of that aspect of our project.
It sounds like you have next steps then. That sounds good, right? Okay, so I'm sorry, Phil, we have to actually move to the next team, but they will be at the gallery show tomorrow, so we hope you all will join us and ask them more questions then. Thank you, Microbiome Repopulation Project. Wonderful job. We're going to move on to New York University.
Our current geological age, the Anthropocene, is marked by technology-enabled geoengineering, with human agency over the environment causing a substantial impact on Earth's geographies, biological species, and ecosystems. For the Biodesign Challenge, we are speculating what might happen if human agency is relinquished to non-human organisms, and what effects it may have on the environment. Our speculative project Immersa, the all-purpose microbial service agent, is a line of technological devices that provide human-scale agency to microorganisms. Why microorganisms? They make up the majority of the living species within our planet. They predate any multicellular organisms, and they are necessary in all ecosystems studied to date. What is human-scale agency for a microbe? Since microorganisms exist at a micron scale, we designed a human-scale robotic interface that can perceive microbial signals and act accordingly. Basically, a microbially controlled robot. This robot can intervene in the built environment with the same impact as a human being. I'm Shireen.
I'm Erin.
And I'm Gopika. And we are the Immersa team. To begin, we began brainstorming with worlding exercises. We realized that there are many microbial communities to serve, so we decided to start at a smaller scale, beginning with the plant-associated microbial species. We focused on seeing a world where the power hierarchy between humans and non-humans are challenged and broken, and a future wherein the planet was changing from human-made damage. We reimagined how technology would work for microorganisms to provide agency for them to reintegrate and to provide them with more possibilities of redesigning the spaces they inhabit. We storyboarded a speculative device that would be adaptive to their surroundings and environment. This device would be able to serve plant microbes to facilitate and expand possible options of regrowth, evolution, and adjustment on a larger scale. We speculated the impact of what this would look like at an architectural scale in different environments when the plant microbiomes of these regions used the device. We looked at metropolitan cities, Superfund sites, ghost towns, and sites affected by nuclear disasters. Using these sketches, we narrowed down the functions of the product, the primary functionality being to provide microbes with the ability to redesign the spaces they inhabit.
In order to shift agency towards plant microorganisms, we proposed an up-to-date Immersa model with the following characteristics and functions. Disclaimer ahead: to better introduce functions to the human audience in these pictures and videos, Immersa's interface has been translated to English, and this language system is not known to be related to any of the microbial communities. The functions of Immersa are: firstly, it provides detailed analysis of environmental factors and sensors enabling detections, such as light, humidity, water, geography, air quality, toxin levels, and possibly activating long-distance signaling and predictions to help indicate a higher survival chance. Secondly, it perceives information from plant microbe interactions and ultimately microbes to Immersa interaction to keep track of a flow of messages by mimicking the bionic communicative system as a spontaneous method of a communication channel, digitally built by materials that speculated with plants and microbiotic dataset. Therefore, it also comes with a communication system that is specifically with microbes, fulfilling needs based on data calculations and simulations, provides a reductionist approach by which both plant and microbial ecophysiological responses are measured, and they are explicitly considered over time and environmental conditions so that it can efficiently provide resources and assistance if needed, and they are always ready to listen to the microbes' requests. Then it leads to an essential function, which is to act on these incoming requests. And these tasks involve displaying analyzed options that fit the best interests of microbes, such as scenarios like planting the designated seeds and plants in certain locations if the microbes agree to accept one of the options. And being able to plant, the housing incubation is also speculated in used for transportation and behaves efficiently for microbes' needs. Finally, we want to include the possibility that it could potentially be a network system for other species, enabling communication and assistance further beyond plant microbiomes.
To better visualize the functions and mechanisms of Immersa, we created a first-person animation of Immersa operating in a metropolitan environment. We understand that microbes do not communicate in English, so this animation is translated into English for the human viewers. Immersa begins by forming and establishing connections with a microorganism through its built-in communication system, which allows the Immersa to receive individual input requests. Immersa takes the data and analyzes the information to create options for the microorganism to choose from and create a plan. Once the microbe's plan is finalized and confirmed, Immersa will facilitate and execute the request plan. In this example, Immersa is planting 100 Herberaturia fusarium plants within its movement range.
With emerging concepts involved with nanotechnology, nanomaterial-based biosensors, and existing big data infrastructure, which is a method for analyzing large-scale NGS datasets using computational algorithms, statistical tools, and supercomputers, and most importantly, the machine learning and deep learning process. The function of establishing a communication system through microbes can be considered feasible and adaptable in a possible future where signaling information and the communication dataset are directly transmitted from the microbes with less human intervention. Through indication of research, we are also seeing a pattern of plants that are actively seeking sorts of microbes as a way to suppress soil-borne pathogens in order to survive. And thorough common usage of microfluidics, which serves as an inspirational method, may potentially help Immersa to understand plant microbiome interconnection better, and most importantly, learn the changes in the core microbiome and what the recruitment process is like, to further enhance the process of providing the most efficient help to the dedicated microbes.
As a speculative project, there are many unknown factors in the prototyping process and the effects of Immersa. Exploitation. As a device that roams freely in the environment, we are unable to predict if people will take and exploit Immersa for personal or unethical reasons. Balance. Immersa only facilitates the needs of the microbes. As a result, the impact and interactions towards other plant, animal, or microbial species is unknown. Thus, the current world balance may change drastically and could result in a complete imbalance. Interactions. As every circumstance is different, we do not have a clear understanding of how microbes interact with each other and how they might interact with MRSA. Materials and human intervention. Since MRSA is currently a speculative project, we are unable to determine what materials would be needed for the actual robot. The battery and power supply of the robot still needs to be determined, and human intervention may be needed for the robot to recharge or to have parts replaced. Priority. There is currently no method of prioritizing the microbe requests. We don't know if the request should be based on analysis or if it should be a first-come, first-served process. Ethics. Although we are interested in giving microbes agency, we are unable to communicate with them. There is no realistic method of knowing if microbes want a service device like MRSA.
Due to these concerns, we propose the idea of using a digital twin, a virtual representation that would simulate how these devices would work using real-time data. Machine learning would help in narrowing down to an iteration of MRSA that would be the most effective when used by microbes. These digital simulations can help experiment and facilitate these obstacles before the creation and deployment of a physical product. Using a digitally simulated environment, we can also observe how MRSA will affect different habitats. Research shows that microbial communities are capable of adapting to environmental changes and differences, for example, fluctuations in the chemical composition of the environment. Their experience will be decided by various factors such as the environmental conditions and the microbial makeup of an area. Thus, each MRSA will look different, as each MRSA's experience will be unique. Here are some possible futures we imagined. We wanted to note that, as human creators of this project, we can only approach these speculations from a human perspective, and we are unable to foresee the possible futures that microbes would want. Thus, these images only reflect our imaginations. However, microorganisms may not create futures that are utopic for other species. Through our speculative project, MRSA,
we hope to imagine a new geological age
where microbial and multi-species agency is possible.
Wonderful. Great job, NYU. Beautiful illustrations. Justin is ready to go. Please.
Thank you. Thank you, team. That was really great. And I think you explained quite a complex idea quite clearly through your video. So well done for that. And it was very brave to take on the topic of the kind of non-human agency thing for lots of reasons. I think you also touched on that really well in your kind of concerns section. So well done for giving a lot of depth of thought for that. My question is, you obviously, there is obviously a level of human control. So I'm wondering if you've really thought about and imagined that future and who those people are. So obviously, you know, they need to be programming and servicing and managing these robots. Who are these people? Are they government? Are they corporations? Are they individuals? And what kind of skills do you imagine them needing to have or backgrounds they come from? Thank you.
Yeah, I can take that question. So we were kind of thinking of this project being people that are very invested in this kind of topic as well, who also believe in spreading agency around to other species, because there are a lot of large corporations specifically that are very human-centric. And so we wanted to kind of collect a group of people that would be interested in kind of shifting agency to these other species. And we thought a lot about how we would need other types of people or professions to be able to fuel this project. There's a lot of technology and research that kind of goes behind it. And so we were thinking of having consultations with these types of professionals and kind of administering that through this digital twin idea, where we would be able to kind of facilitate and create this project through a digital environment. So there isn't as much kind of investment needed in the very beginning. We can kind of have a proposal, kind of formulate a plan before we kind of send it off for it to be created and made. And so we kind of just, there's a lot of consultation that is needed there. And this like prototyping process is like much further in the future. And so like a lot of these functions and unknowns would need to be figured out and individually kind of talked about with different professionals.
Thank you.
Cool. Let's move to Doug. Please go ahead.
Hi, thank you for your very interesting video and beautifully produced and gave me a lot to think about. I have a couple questions about the mechanism you're choosing to communicate both this idea and also the robot itself. So as a human, I have to tell you, I'm a bit put off by this. I'm not excited about the prospect of giving over some of my own agency to robots or even to any other mechanism. So I'd like to know why you chose to use a kind of robot vibe, I guess might be the best way to say this, to sell me this idea. And then if you are using robots, how are you going to convince me to maintain them and to keep them alive? Thank you.
Yeah. So I guess initially when we were starting this project, we were trying to think of a way where humans and other species can kind of live symbiotically and kind of operate on the same power level, especially where a lot of agency is kind of done through humans, especially through technology. A lot of like the geological like destruction and like infrastructures are made by humans. And so we were wondering if there was a way where different species, specifically microorganisms, would be able to have their own agency, and we would be able to ultimately communicate with them and form this kind of relationship that we don't have currently, and be able to work together to live symbiotically. And this kind of idea, we were trying to gear it more towards for the organisms. I guess we didn't really think of it as something that was for humans. It was just kind of something like, oh, maybe we could provide, well, not so much provide, but shift this agency towards other species and allow them to have like these opportunities that can be done through technology. And we wanted to go towards a technological route because at some point it can be a most hands-off kind of solution. We can kind of create this robotic structure and also have this software implemented to where it can learn over time from the environment and these microbes and have it be able to communicate with them. So we wanted to minimize the amount of human interventions as much as possible. So it's really a service device that are for these other species and not so much for humans.
Thank you.
Thanks, Doug. Let's finish up with Phil. Go ahead.
Thank you. I have a question going on that topic of agency. There's lots of different organisms and they all have different kinds of levels of agency. As a human, I have a certain level of agency that's different to a fungus or a mold spore that's floating around this room or the plant that's over there. So how do we decide who has more or less agency by adding technology to facilitate that?
I guess this device is kind of there for these microorganisms, but they have the option to choose to use it or to not use it or to completely reject it. So it might be something that is completely unused by these microorganisms because they aren't interested at all. But I think it's just a possible way that we could shift agency. So yeah, a lot of that is kind of undetermined. And it's really depends on the wants and the needs of these microbes, so they can have the option to choose to do something or they don't have to do it at all.
Yeah, because like every circumstance is very different. So we said in the video that we don't know like how exact mechanism that this device can interact with the microbes. Each microbes are also very different. So we can only, based on the existing field of technology, like molecular biology and also theories like plant neuron biology. So like indications of plants that are made, like they have behavior and they are coordinated by some type of integrated signaling interaction, so that this device can pick up this signaling and then also learn better before they can communicate. Yeah.
I guess adding on to that, we wanted to kind of examine different degrees of impact in the world. So right now, humans have the most, at least from our lens, that humans have the most impact that we see in terms of what's happening to the environment. So we wanted to kind of speculate what it would be like to have microbes have that level of impact in the environment. So that's how we kind of began with this project.
Awesome. Thank you for your question. Thank you for your answers. We are short on time. So thank you, team, for challenging us to use our imaginations. We're going to move on now to the University of Toledo.
We are envisioning a future with a greener way of reducing light pollution in the biosphere through assistance from the surrounding flora. We present our vision, and this is Team Lumen Arbor. Since the beginning of humanity, we have been trying to harness the power of light to navigate through darkness, whether it be by the natural light of the moon and stars or by a material flame. Ultimately, as humanity has progressed through history, we have created an enormous amount of artificial light via electricity, which has increased the standard of living across the globe. However, electric lighting has created another set of problems for humanity, despite also being the solution for many other issues. Now, there's an abundance of artificial light, which is unintentionally illuminating spaces across the globe where it is not wanted. This phenomenon is referred to as light trespass, which can disrupt both natural and artificial processes. Another issue caused by artificial lighting is sky glow, which is excess light that is shown into the sky and then diffuses from the atmosphere into areas that would otherwise be dark. Collectively, these effects are referred to as light pollution, and it is such a large problem that it can be viewed from space. Light pollution is not just a nuisance, but rather sometimes it is a hindrance. Too much light and glare can prevent us from seeing what is right in front of us. And that can be disastrous if the light is concealing a potential threat. Other more subtle dangers that could be caused by light pollution are increased cancer risks from the lack of melatonin production, circadian rhythm disruption, and sleep cycle disruption, just to name a few. When light pollution is present, stars in the night sky are much less visible, stars that have been important to humanity for as long as we have existed. We use them for navigation, spirituality, and to tell time, so losing sight of the night sky is like losing a piece of ourselves and our history. An unpolluted night sky is something that our society has taken for granted for a long period of time, and it would be a great benefit to us all if we could see more of it again. Unfortunately, we aren't the only ones impacted by our production of artificial light. Nocturnal animals, plants, and many life cycles rely on proper day and night alterations to function. For example, baby sea turtles use the light from the moon to guide them to the sea, but with light pollution from nearby human developments, they often head in the opposite direction, which in most cases leads to their unfortunate demise. This is one of many ecological disasters caused by light pollution, which may have many long-lasting effects. Despite the damage humanity has inflicted upon nature, we believe that nature itself could have a solution that offers us enough light to see without disrupting any other process, and that solution could be bioluminescence.
Bioluminescence is a natural phenomenon that causes organisms to produce their own light. This adaptation has independently evolved more than 40 times in a variety of aquatic organisms, such as deep-sea fish, bacteria, insects, and fungi like Neonathoponus nambi, otherwise known as foxfire mushroom. Bioluminescence can have a multitude of functions such as mating rituals, deterrents, and prey lures. In fungi, it is speculated to be the byproduct of their metabolic process. Bioluminescence involves the production of light by a living organism via a series of chemical reactions. Depending on the species, the method used to produce this light can vary, but the component that remains consistent in all methods of bioluminescence is the use of the enzyme luciferin, a light-emitting pigment. Specifically, we are interested in fungal bioluminescence, which utilizes a chemical reaction of luciferin and luciferase that together produce this form of light energy.
Humanity has long been enthralled with bioluminescent organisms, with the earliest records of them dating back to 322 BCE by Greek philosopher Aristotle. Interestingly, on the first submarine used in military combat known as the Turtle, foxfire bioluminescent mushrooms were used to illuminate the needle on its barometer. Once a submarine dropped below the reach of sunlight, this was the only source of light available on the craft. In more recent decades, bioluminescence has been used in multiple scientific applications. One example being the use of GFP, or green fluorescent protein, from jellyfish. This has been used in medical lab settings as a biomarker to track the active progression of cancer cells under black light.
Brilliant minds across the world have illuminated the possibilities for bioluminescent projects.
And some of these include Teresa Van Dongen's Ambio, a visualization of how to use nature as a source of energy back in 2014. The project features bacteria that naturally luminescence when moved, so the lamp turns on with the slight push. The end goal is to create an installation that shows the environmental and aesthetic possibilities of bioluminescence. Karen Sarkisian, co-founder of Planta, also highlights that the luciferin 3-hydroxypidine is stable and cell-permeable, and that Neonathopanus nambi, a bioluminescent mushroom, is roughly as bright as a firefly.
So how can we apply the light-emitting mechanism from bioluminescent organisms to solve light pollution and access to light around the world? Our solution is to create bioluminescent trees using luciferin from fungi. The luciferin would be integrated into the metabolome of a tree. What this means is that the fungal bioluminescence pathway would be integrated into the caffeic acid cycle of the tree. Caffeic acid is part of a tree's metabolic pathway, and certain fungi have the ability to convert a caffeic acid molecule into luciferin to create bioluminescence. Once this occurs, genetic engineering techniques can be used to insert the genes from the mushroom into the genome of the tree to create transgenic trees. Similar approaches have been used to create bioluminescent tobacco plants. Scientists have inserted genes into the tobacco plant that encode certain enzymes involved in the fungal bioluminescence pathway. This process gave the plant the ability to produce light. Similar experiments were done in other plants such as tomatoes and dahlias, and the results showed success.
One concern with genetically engineered trees is the possibility that they could harm wild tree populations through cross-pollination or becoming feral and disrupting local ecosystems. For this reason, we've decided that a sterile tree is necessary for this project. Our team did research on the types of trees that could be sterile, and we came to the conclusion that seedless fruit trees could be a great candidate. In particular, we are interested in the Spencer seedless apple tree for its infertile flowers that produce no petals, no pollen, and typically lack stamens, the male part of the flower. Apple trees have a few advantages. For one, they're a common plant in lab studies, and their mechanisms are well understood. In fact, they are the 10th plant to have their entire genome sequenced. And secondly, they can grow in a wide range of climate zones, specifically zones 3 through 9, so they will be available in most locations with proper care. Of course, we do not have to be forever limited to this tree species, but for the sake of simplicity, this is where we plan on starting. In order to create enough individuals, these trees will likely be multiplied artificially through common propagation methods such as grafting, which involves taking part of one plant and physically attaching it to another plant, or through cuttings, which involves taking part of a branch or root and allowing it to grow in water or soil until it forms all the necessary components to be planted as an independent tree.
All of these bioluminescent projects have been in discussion for the past decade. Attempts have been made to boost the luminosity of the glow we see from the bioluminescent plants, but there has not been progress so far. Given that others have not been able to figure this out yet, how will we boost the intensity of the light in our trees? Using the process of chromosomal integration of genes with multiple copies, we would manipulate the phenotype of our inserted gene and place it in the tree's metabolome. This process may be problematic, however, because it is not always stable. Despite the current instability, we project that using this method will allow us to directly optimize multiple copies of the luciferin gene and metabolic pathways, which in our case is the caffeic acid cycle. In case the chromosomal insertion does not work, we have come up with an alternative solution. Integration of the target genes into the host chromosome is a preferable strategy to overcome the drawbacks of plasmid-based overexpression. However, this often requires iterative rounds of engineering because many current insertion approaches are limited by the size of DNA that can be transferred onto the chromosome. Construction of highly engineered strains is very time consuming. There's a solution to this as well. A series of knock-in, knock-out vectors can be constructed for integration of large DNA sequences onto the E. coli chromosome and sugarcane smut fungus at well-defined loci. These kekoplasmids target 3 non-essential genes/operands as insertion sites, and from there we can have multiple copies of a fungal luciferin inserted that will express themselves in mass. However, All attempts to create bioluminescence in plants has resulted in a very faint light. So it will be necessary to pinpoint how much light these genetically engineered trees will produce and how to increase the light intensity if necessary. And from the recent studies in Rambouillet, a small village southwest of Paris, it has been estimated that if a plant could produce, they would emit 57 lumen of light each. We would need at least 40 plants in every 30 meters on each side of the road, and that's equivalent the lowest class street lighting required on road by pedestrians. We aim to produce bioluminescent trees that will have enough light to illuminate the pathway you walk on, eliminating the need for the glaring street lights. Our bioluminescent tree will be having a glow equivalent to that of a 40-watt lamp, giving 360 lumens. This new brightness level is in stark contrast to the normally up to 5,000 lumen in residential areas and a staggering 18,000 lumen on roadways. Our bioluminescent trees also take into account the adjacent thriving ecosystem by emitting light at a frequency of 550 nanometers, which is green light. Plants reflect green light so they should be blind to the light produced, and this wavelength has a smaller impact on wildlife than traditional lighting wavelengths. Green light is also at a wavelength that does not disrupt night vision like white light does. So night vision could also aid in people's ability to see at night in addition to the tree's glow. We have also considered the factors of nocturnal, diurnal life cycles of small plants, feeding patterns of insects that pollinate the flowers of surrounding flora, and found they are not at risk either.
Now, how can we build off of these projects to create the world of tomorrow? Imagine a future where you're walking down a path illuminated by glowing trees. What would this world look like? Would our everyday lives change, and would light pollution affect us in the ways that it has been affecting us now? Furthermore, how could it change the way that we interact with the environment? We are envisioning a future where bioluminescence can be utilized to light our world. Within it, glowing trees will replace many of the street lamps in our neighborhoods, on small streets, anywhere else that people seek light. The glow will be soft enough that it does not disrupt natural processes and bodily functions, but still bright enough that you can find your way. As of today, we overuse light. Often, lights used to light up residential areas, shopping centers, and small streets are far brighter than necessary. And that presents one of our largest hurdles, which is retraining ourselves to not overconsume light. To kickstart our idea, we hope for museums and attractions to spread the awareness of the power of bioluminescence. These spaces will feature various bioluminescent organisms like bioluminescent mushrooms, fish, dinoflagellates, and small plants to curate an interactive educational experience for the public. Here, the public will learn about the impacts of light pollution on themselves and the natural world, and learn about alternative sources of light. The first bioluminescent tree prototypes will be housed in these locations for the public to see and experience. These attractions can also boost the local economy by encouraging tourism while spreading educational information and sparking curiosity and appreciation for the natural sciences. These sites will hopefully open people's minds to the idea of utilizing bioluminescent public spaces to lessen light pollution production and reimagine what our world could look like.
You are taking a stroll down a sidewalk near a park in a metropolitan city. Imagine the glaring city lights are replaced by the soft luminescence from our bioluminescent trees. This is the future we are imagining for generations to come.
Our next step into the future is at our doorstep. But what about the bioethics involved with the trees and genetic engineering? If we can modify the nature around us, will it also affect the small ecosystem thriving around the luminescent trees we will be working on? Genetic modifications have been debated for some time now, but we should remember that through this science, we have been given seedless fruits and better crops that are disease-resistant and more nutritious. What are the ethical and moral implications that we might be facing by creating these bioluminescent trees? The presence of these trees will raise many philosophical and religious questions, in addition to requiring some amount of governmental regulation. These trees could also cause concern for those who believe that nature should be left untampered. We think as a team that these concerns are actually a hopeful byproduct of the tree's existence. The glowing trees will create a conversation and debate about the role of nature in our lives, and potentially increase an appreciation for the power of nature to provide us with the things we need, including life.
It is our goal to simultaneously provide enough light so that the world's cities, towns, and villages can have enough light to see at night, but do not disrupt nature or our natural cycles. With further research, we can see our world and solve the issues that bright lights cannot. We can chase away the dangers of darkness without becoming a danger to ourselves.
All right, thank you, University of Toledo. We'll start with Bill, please.
Hi, thank you very much for that interesting concept that you have there. I have two questions. The first one is, what do you think might happen to things like birds or possums that live inside the trees when they're glowing at night? And secondly, maybe I'll come back to that. I've forgotten what I was going to say. So maybe just what might happen to the animals that live in or eat trees, do you think?
I can take that, and my team can add on that. So, to answer your question about possums and like birds which are flying nearby the tree, so we have chosen seedless apple trees for a reason because apple trees have certain habitats, certain places they grow in, and we have tried to study the type of creatures like birds, possums, even ants, like the smallest of insects to the largest of insects. How are they affected by that? And since the light emitted by our bioluminescent trees is green in color, and the wavelength is not that long that can affect the vision or the life cycles, the creatures around. So that has been taken in note, that has been noted, and we have been working on that. And they would not be affected by it.
If I might follow up with the question that I had, which set my mind, why not just turn some of the lights off?
I believe there's not going to be any less of light in the future. If we consume light is for better resolution, but with the coming higher resolution, we are getting even lesser sensitive to the people and the species around us. It's not about the light we consume, it's about the sensitivity to species around us as well. We can consume less light, but for a better resolution, we can just substitute it for a better light and keep in note of the species around us.
Yeah, and I'd also like to add that it would also increase the green space in places where people live.
Mm-hmm. Great. Todd, please.
I wanted to maybe unpack a little bit some of the societal sort of questions that you touched on a little bit at the end there. And I'm curious if, if by doing this, by introducing all of these trees, if you are swapping one pollution for another. And, so one question is, do you see this as introducing genetic pollution into the environment by swapping out the natural light for the genetically engineered light? And then have you thought about whether or not society is actually going to accept all of these new genetically engineered trees, replacing, light poles and those types of things?
To answer like the first half of the question, we don't aim on expanding the trees to make every tree bioluminescent. We aim on making only a certain trees in certain areas bioluminescent so that we don't spread the idea of even green pollution in that way. We already have light pollution, which is sensory, and we don't want to make it even more, even if the color is changing. And to answer the second half of the question, the society, it always takes time to accept changes. It grows on, and once it's introduced to museums and attractions, it's going to take a while. And as a first prototype is introduced, and after years, maybe we can move on to something bigger, and society may start accepting that we do have sensory pollution.
Yeah, and that was also one reason that one of our goals was to create trees that would be sterile, is so that they couldn't cross-pollinate and introduce like mutant genes into wild tree populations.
All right. Thank you, judges. I'm sorry, Eve, we actually have to move on. We're very short on time today. Wonderful job, students. Those are some tough questions. We're going to go into a break now for about 15 minutes. Please stay with us, everyone. We have some videos from people in our community, artists, designers, and scientists, and some of our sponsors. So we hope you stay with us, and we'll see you in about 15 minutes. Thank you.
The mud is electric. You're stepping on it, underneath your feet, in your backyard, or the swamplands near your home. The mud is electric. The mud is everywhere, and it's hiding a life form with incredible potential. Bacteria. Amazingly, some bacteria have learned to thrive in anaerobic conditions, especially Geobacter sulfuridescens. They can build thin threads through the soil, protein nanowires that carry current up towards oxygen to metabolize. What does this mean? To get to the point, Geobacter can generate continuous electrical power from the air. The implications are titillating. Fashioned into a film, these electrically conductive protein nanowires generate electricity when a moisture gradient develops. We talked to Dr. Lovely and Yao, the core scientists involved in this 30-year research. They told us, properly assembled, such a film could actually power a cell phone, and the nanowires can easily be produced in bioreactors through a cheap and low-impact process. As designers, we marveled at this, and of course, we sought to bring it to life. We are a global team of artists, designers, storytellers, and researchers. Our goal is to engage in new, meaningful connections with our electronic companions through planet-first design practices and nature-based solutions. It's no secret the model around our electronics is obsolete. From mining to energy use to e-waste, the impacts are too grim. Around 7 billion people own devices, and while they're radically more efficient than a decade ago, their sources of power are not. The power experience and battery technology have failed to meet our needs. But what if we could design away from batteries? Change the charging experience? What if we could power our devices in the most local of ways? From the air, right there. What if we created living skins for our devices? Geobacter inspired us to think deeply on this and apply it to the speculative phone, the Pixel Bio. But we needed to create a skin with enough surface area to power a heavy-use device. There again, the inspiration came from nature. By printing the skin into the shape of human small intestine villi, we can achieve a large surface in a very small space. The calculation is simple, but we'll spare you the details here. All you need to know is that it's entirely possible to power a Pixel Bio that needs 5 volts and 1.2 amps to charge, using a total surface area of 2 square meters. And to achieve this surface into the compact space of a Pixel Bio, we mold chitin into a phone case shape enhanced with synthetic villi. That's the cradle for the Ballerian circus that animates the device. In parallel with the bioreactor, we can then produce the nanowires and spray them into a solution onto this chitin base. And to create the moisture gradient we need to activate the nanowires, we dip this assemblage into a porous, conductive, hydrophilic alginate solution embedded with gold wires. These are the electrodes that direct the current. Made this way, the skin will power the Pixel Bio at average ambient humidity, 40%. That's likely the air you're breathing as we speak. It of course works in other humidities too. On top of that, alginate and chitin, the materials we mentioned above, are abundant, renewable, naturally occurring resources, with the potential to regenerate marine ecosystems and provide livelihoods for coastal communities. But self-charging skins could be created out of local species, using regionally specific manufacturing practices, and allowing for local population to use place-based resources using our open source process. The impact? No more charging cable, and a smaller and longer lasting battery that recharges ad infinitum. And as a bonus, a skin that biodegrades back into the earth and supports local economy in the making. Aggregated, that's less demand on power grids and much less e-waste in the landfills. And the potential of this technology is just at its beginnings. Scientists are finding new species of microorganism that can generate even more voltage, and bioengineering promises to increase their efficiency further. The direction of the nanowires in this film can also be tweaked to enhance power generation, and the solution in which they float can heighten conductivity. The next 5 years will allow us to prototype and iterate on the already well-developed experiments of Lovely and Yao, as well as explore these enhancements, the manufacturing process, and of course, answer some of our remaining questions around the layering and lifespan of the skin. Future research could eliminate the need for a battery entirely. And there's a deeper impact. The self-charging skin also ushers a new materiality for our devices. Now our phone is textured, layered, deep, soft, touchable. By holding it in the skin, we shift our relationship to our electronic symbiont. It's there to care for. Now our perception towards the devices moves from disposable to companion species. Its end of life is no longer in the landfill. It's back in the mud.
To those who care. What if we could turn flowers into coats, air into ink, grapes into leather? Let us introduce you to the greatest inventor of all time, nature. And us humans as its best creative partners. Because our actions now will define life on Earth for the next 100 years. This is why we innovate. For a planet where both humans and nature thrive.
Intertidal Wear by Alt Labs looks to living systems as a model for closing the circularity loop. Designing for living systems is all about the system itself. Better understanding of the system results in better alignment with it, resulting in less leakage. We propose a device subscription model that is access-based and offers consumers a way to engage with new materials and emerging biotechnology. This system provides an intuitive framework of clear methods for disposal and interaction within a tiered subscription model featuring multiple consumer entry points. By leveraging the qualities of a living system, we are designing leakage out. In this model, consumers will clearly understand device collection and materials retrieval such that valuable materials remain in the system itself, not in a box in the attic or the landfill. Building from the insight that consumers love their data more than their devices, we realized it was possible to create a system that consumers could relate to, engage with, and ultimately fall in love with. Much of our own behaviors are inspired by our own intuitive understanding of our place within systems. We don't put batteries in the trash. We know they need to be collected so they don't contaminate our planet. We take special care of these types of objects not for our love of the thing, but for our love of the system. The cloud is an apt metaphor for the way in which we currently understand data storage and access, a nebulous vapor with very little points of entry for the consumer. This metaphor fails to convey how people fit in. The Tidalware system creates a more relatable transitional space, better bridging the worlds of devices and data. The intertidal zone is where ocean and land meet. Composed of areas defined by the amount of air or water they are exposed to, this area is characterized by an abundance of specialized organisms, is biodiverse and highly interconnected. This is a fragile yet resilient ecosystem that relies on nutrients provided by the constant ebb and flow of tides. It is also an ecology that has nourished humans for thousands of years. This transitional zone provides a physical place where uniquely specialized organisms, adapted to completely different worlds, occupy a single space. The interaction of land, sea, air, organisms, and humans perpetuates the tidal zone nutrient cycle. Each zone provides essential nutrients to the other. This model for circularity provides the foundation for our access-based concept. Each zone is home to something unique: starfish in a tidal pool, rocks for climbing, seaweed in the tides. This tiered experience provides a layered and engaging visit. The tidal zone establishes a clear transitional space. The ocean as our sea of data, land as consumer access, devices bridge the gap in the intertidal zone. We imagine this taking many forms: a physical booth at a Google store, live online, or a pop-up shop. The metaphor makes the system work. The concept could play out in a multitude of forms. The literal zone metaphor builds on our pre-existing understanding of cycles and grounds an intuitive understanding of a new Biodesign Google hardware system. The consumer will lease devices in a subscription model. Devices will be understood as temporal, constantly cycling in and out of use to be reused, updated, and modified. To withhold a device in the attic or in the landfill would be in clear disharmony with the cycle. All devices will be owned by the parent company. The consumer is ensured they always have the newest updates and latest in biotech advances. There is incentive to bring devices back to the parent company for matriculation into the device nutrient cycle. In the one-on-one package, the user subscribes to data using a single device. During the membership, the device may change. It might be replaced, upgraded, its parts might be recalled for other use. But the user subscription and device access remains in place. In the ecosystem package, consumers subscribe to data and to a product ecosystem. Entire households enter the closed loop of both data and electronic components. Over time, these components would cascade from one device to another. These devices are fed back into the system to be taken apart for resource retrieval and reuse in new products and upgrades. Over time, a user may decide to add or subtract more devices to their plan. The main idea remains: no matter how a user chooses to engage in the Tidalware system, they are always in the system. There is no incentive to keep a device under a bed. All devices are meant to support each other and be reclaimed by the manufacturer for ultimate control over true product circularity. This is a user-accessible system with many points of entry. Based on the multi-level points of interest in the intertidal zone, this space features levels of engagement. Cascading parts displays, emerging biotech exhibits, a place for skill shares, and a workspace where visitors test new components. All of this creates a community space, inviting conversation, innovation, and original thought within biology and hardware. Visitors may encounter information about a new mycelium computing system, learn about algae batteries, or self-healing hardware. This type of access to emerging technology inspires new application, collaboration, and innovation in the field. Entering the space, you might linger at different experiences or head straight to a help desk for upgrades or subscriptions. This place, physical or digital, permanent or pop-up, would have the feel of a genius bar crossed with the Natural History Museum at happy hour. This is a lively space of sharing, experimenting, conversation, and commerce. All of this comes together in a system we can fall in love with. Intertidalware is engaging and encourages strong new behaviors, heavily rooted in pre-existing truths around nature's interconnected living systems. Intertidalware by Alt Labs.
Hi, everyone. Welcome back to our last session of day 2. We're going to jump right into it. So the first video we have for this session is University of New Mexico. Please take it away.
Hello, everyone, and welcome to Algae Flow, a meditation, an exercise in design philosophy and creating a symbiotic system for algae and human well-being. My name is Blaine Greiner, and I'm representing the University of New Mexico at the 2022 Biodesign Challenge. So the project originated when I received an unexpected sample of free algae from my biodesign class at UNM. I decided to undertake a design process which was informed by learning about the algae through direct observation and care, and take these learnings and come up with a design project out of them. My initial objectives were to create an exercise in using meditative, intuitive, and responsive design practices when working with a biological medium. I wanted to let the initial symbiotic learning process reveal opportunities to address ecological and individual well-being, or, in other words, doing it backwards. I knew I could count on my past experience in indoor growth systems to help me out with this project. I have had prior experience working with aquaponics, a symbiotic system for growing fish and plants to create food for human consumption, and using some of the aquaponics water, I did create a sunlight-based algae system in the past in order to try to process the aquaponics wastewater into something useful. Aquaponics system. And now I'd like to discuss the design process that I used. So my first step was receiving an unknown sample of algae on February 8th, 2022, which I believe to be chlorella. And then I spent several weeks observing the algae's reaction to various changes in light, temperature, and various methods of air exchange. In addition to general observations, I noted that the algae flocked to the bottom of the sample tube in a high heat. My experiment was off to a good start. The second step was creating an initial growth scaffold design. Based off of my initial observations, I designed a growth scaffold with integrated air tube, which you saw in the previous video, which aerates and agitates the water while providing surface area with less agitation for algae colonization. I use parabolic shapes for their flow properties, rocks and rivers, bird beaks, biodynamic flow forms, and I thought that the bubbles moving over the parabolic shapes would agitate the water to both expose the algae to air and also provide safe spaces for it to grow. My next step was to undergo a second round of observation and culturing. I increased the container and grow scaffold size, and I further observed the algal culture for an additional 2 months. Much to my surprise, the algae seemed to be growing in a manner which utilized the growth scaffold and is impacted positively by the initial design assumptions. Now, after working with the algae for a period of approximately 3 months, I started to think about what I could do with it. So I started an initial prototyping phase where I designed a 3D printable enclosure which can be modified for different purposes. Initial designs included a bioreactor with algae growth scaffolds and a flow wall for aeration and piquing human interest. While having a totally modular algae growth system was a great idea, I found it challenging to implement because it was too broad. So I'll chalk it up to a thought exercise and possibility. Using the learnings from step 4, I decided to take my thought experiment for a modular algae system and focus in on some designed objectives, uses, and a problem to address, which I will be showing in the next few slides. And from that, I created a small model for a room-size algae growth system installation. So, after going through this design process and working with an algae specimen and growing with it and learning with it, I decided that a problem that we could address collectively would be that the industrial disconnect from the biological world and its rhythms is negatively affecting our ecosystem and impacting human health. So, how do we address ecological well-being and then at the same time address human well-being? So while I may not have the answer to the problem, which is such a big problem, I wanted to come up with a solution that took the design process that I developed with the algae and create something that sort of spoke to the principles that went into that process, as well as providing someone with a viewer experience. So my solution to demonstrate this design philosophy would be developing an indoor installation for algae growth, which would simultaneously purify indoor air while providing meditative sound, sights, and opportunities for meditation and connection with a biological entity. On the right-hand side, you can see the model that I've been making as part of my process to develop a room-size installation of this algae flow system. So thank you all for coming on this journey with me, and hopefully, going forward, we can see the biological entities that we work with as partners in addressing the problems we face together.
Awesome. Thank you, Blaine, for that video. I love the pacing. It felt meditative itself. So appreciate that. Justin, let's go for it. Right into it.
Thanks, Rena. Thanks, Blaine. It was really interesting for me as well, because your name, Green O'Grainer, is the same as a quite prolific scientist, Andre Grainer, who works with algae. So I was wondering if you were related or something. I think he's in Germany, and he has done a lot of stuff around CRISPR and Chlamydomonas algae. So that was quite interesting to me. I think that as someone who, as you know, my scientific background with algae, this was really interesting to me because it really shows me how differently designers approach, algae culture, for example, out of the lab space. And that's really important when we're trying to think of these different forms of design applications like a meditative thing. I loved the final design, and I think it was very clever as a first iteration. So my two questions were, firstly, what was the kind of green fluid coming through the process? Because that's obviously not the algae. I think that was a wise choice for numerous reasons. And the second one is, how are you refreshing the culture? Because it will obviously become too dense at some point. Those were my questions. But thank you. That was a really great project.
Oh, thank you. Yeah, great feedback. So the first, I used just green food coloring because I didn't want to subject my algal culture to, I didn't know if it was going to work or not. And so it was kind of just like a first try experiment, but I wanted to capture it at the same time. So I wanted to kind of going along with some of the themes that I was exploring in this project. I wanted to kind of try it first with water or something that wasn't, my co-conspirator, and see if it would work. And so eventually I'll probably introduce the algae into it and see how it works. And then as far as refreshing the algae, I'm still learning about it. I feel like there needs to be kind of a bottom reservoir or something that also kind of filters and sort of refreshes it. And that was part of the thinking that went into the initial sort of idea for a modular system, was to have an area that could filter it out or harvest it for food or something like that. But it would probably be a reservoir underneath the installation that would have this sort of filtering mechanism in it. And of course it would be human involvement. So maybe it would be me being actually with the installation for the whole time, and also taking care of it and kind of being in community with it.
Yeah, great. I think there's so much literature in the science community about different ways of culturing algae that you might find useful to draw from. But at the same time, I kind of like that you've gone through your own methods, because otherwise, you've come up with a lot of new things as well. Really great. Thank you.
Thanks, Justin. Let's go to Phil, please.
Hi, thanks for that interesting presentation. I was wondering if there was any specific reflective or meditative practice that inspired the project.
I've been reading a lot of Zen writings the last I think 5 years or so, and so there's but I also have a background in teaching and being sort of an organic farmer, and so this whole process of staying with something and grow and working with biological entities for a long period of time, not necessarily, not as long as some people, but for me it usually was annual vegetable crops. So this idea of kind of working with something over this long period of time and having this sort of continued relationship with it over time was so kind of a combination of that feeling, and then sort of the sort of Zen philosophies of being present, and sort of this idea of beginner's mind, and kind of going with a fresh approach to something constantly and seeing what comes out of it. And even if you do know a lot about it, sometimes pretending you don't know a lot about it and sort of introducing yourself to whatever your practice is or whatever you're practicing, just to kind of get a fresh perspective.
Good answer. We'll finish up with Anya. Please go ahead.
I think there's something powerful to kind of bringing in an open mind, an unbiased mind or untrained mind into, to see new challenges and design solutions. I'm curious, like, you did present us with a proof of concept prototype. If you had like another, 2 months or something, what would you imagine being like your next step? Like, where would you like to see this go kind of like in the next iteration, since they've kind of like changed so drastically in between them?
Yeah, I think part of my process has always been being open to really weird and random changes and kind of taking new information and trying to incorporate it. I mean, what I'd really like to do, I think the next step would be to scale it up and to really like, because right now I've just got kind of algae in a tube and I've got a concept for the system that sort of flows and changes, and to really kind of to figure out the nuts and bolts of that process and how I could like purify indoor air and do all these things while still kind of maintaining this, making sure the situation is beneficial for the algae and for us as humans, and like working on this sort of symbiotic relationship and having this sort of system that goes into the installation be representation of that sort of symbiosis.
Great, thank you.
Awesome. Thank you, Blaine. We're going to move on to the next presentation. Up next, we have the University of Southern California.
Prosthetic bee habitat, growing food networks and communities. Chapter 1. There are no separate systems. The world is a continuum. Donella Meadows. Our planetary systems are facing decay due to anthropogenic forces such as burning fossil fuels, poor waste management, and the destructive practice of industrial agriculture. In Western human cultures, we live our lives in an individualistic, linear timeline. We have structured cities in ways that have caused habitat fragmentation of green spaces and mental disconnects from natural systems. We follow demands for work schedules, often pushing aside the ones that our bodies need to rest and regenerate. This disconnect runs even to our most basic need, food. Seemingly healthy foods are packaged in plastic. When we talk about foods, we usually mention brands and tastes, not the labor, land, and organisms responsible for the production of them. Industrial agriculture has distorted the natural habitat with pesticides, monoculture crops, excess waste, and disregard for ecological health. An essential part of this labor force is bees. They pollinate over 70% of the world's food, a process necessary for reproduction. However, if we look at how honeybees are functioning within our food system today, we find some unsettling truths. Chapter 2. Migratory Beekeeping. Before European colonizers brought the honeybee to America, native bees alone pollinated the wild flowering plants and crops of indigenous people. Today, the agriculture industry sports far too many crops for natural pollination to support. The current model of farming completely depends on migratory beekeeping, where billions of honeybees are shipped around the country to pollinate crops. For each seasonal bloom, these bees are passed around from crops like almonds, apples, plums, and sunflowers. But even in these controlled containments, bees are dying. In the past 10 years, American beekeepers have lost between 30 to 60% of their hives, because packing so many bees close together can easily spread pesticides, diseases, mites, and infections. Migratory beekeeping is thought to be one of the primary reasons that so many honeybees die and suffer colony collapse disorder. Forcing the bees to gather pollen and nectar from large amounts of a single crop also deprives them of the diverse, nourishing diet of wild habitats. This system is unsustainable. In order to lessen honeybees' workload and our dependence on them, we need to revitalize the native bee community. Most native bees are solitary bees. Instead of a hive, solitary bees operate on their own, simply foraging, mating, laying eggs, and repeating. They often live near each other in underground tunnels, holes, in logs, or stems. Because they don't have pollen baskets on their legs, they lose more pollen as they fly, making them around 120 times more efficient pollinators than honeybees. Revitalizing solitary bees will lessen the burden from migratory beekeeping, boost biodiversity, and create a healthier balance in the ecosystem. Chapter 3. Using methods of the prosthetic, a new program can be understood as a new integration within an existing system. Prosthetic Habitat by Malini Leah Famvuti. In order to address the disordered agriculture cycle, we must learn to create new systems. One way humans have attempted to care for solitary bees is by building bee houses. These houses are often wooden boxes with tubes or holes for nesting. We have designed an improved solitary bee house system. Rather than simply providing a shelter, our prosthetic habitat takes a step further in supporting the complexity between its biologically reliant relationships. There are three main parts to our system: bee shelter, bee immunity boost, and soil microremediation. They all work together to mend native bee habitat fragmentation and boost efficient pollination for not only crops, but its surrounding greenscape. The bee house is made of living mycelium, the root-like structure of a fungi. We have chosen this medium because recent research has shown that fungal extract greatly improves bees' immune systems and protects them against viruses and infections. Research also indicates that the bee gut contains beneficial fungi, which assist the digestion of food and may have other protective functions. Bees are naturally attracted to mycelium and have been observed to sip on its sweat, scraping mushrooms for extract, or to take a mycelium bath. By living amongst the healthy growing fungus, the bees' immune systems can greatly benefit. This bee habitat mycelium can be grown with common oyster fungi. Oyster fungi are not harmful to bees and have enzymes that can break down a variety of agricultural wastes. On top of the immunity boost they provide to the bee, the fungus can help degrade biological wastes, chemical fertilizers, and pesticides into less toxic forms using a process called microremediation. For example, heptachlor is a compound used in many agricultural pesticides and has carcinogenic effects on human beings. Oyster mushrooms are one of the most effective fungi to degrade heptachlor and its toxicity. In addition, oyster fungus can also kill varroa mites, which are a huge threat to the bee populations, and solitary bees are often a reservoir for their pathogens. We use the hemp hurd and mycelium blend to be grown in a mold. The structure we designed for the mold fuses digital fabrication methods and biological matter. The undulating surface was generated by a trigonometric function, while the proportions, scale, and base profile were generated by a series of constraints and functional parameters based on the sizes needed by the bee. The overhang created protects the tunnel entrances from wind and rain. The model was built by a CNC mill with a 3-axis robot to create the form and cast in plastic using vacuum forming. Living mycelium is then placed in the mold to take its form as it grows. On the back-end side, the fungus is open to grow mushrooms. With the bottom of the structure placed in the soil, the mycelium is free to expand its network, thus microremediating the soil over time. After about 5 days, the mycelium is removed from the plastic mold, and phragmite reeds are positioned under the form's overhangs as a nesting space. Bees such as the mason, leafcutter, and carpenter typically nest in tunnels of dried plant stems or reeds. For example, after mating in the spring, a female mason bee will find an existing tunnel to nest. She gathers mud to build a back wall at the end of each tunnel. Then she forages and places a nugget of pollen and nectar in the tunnel for food once the larvae hatch. She lays her eggs on top of the food and gathers more mud, plugging the hole and sealing them off from predators. When these new larvae hatch in our bee house system, they will grow stronger through generations with immunity benefits of mycelium. Chapter 4. Life doesn't make any sense without interdependence. We need each other, and the sooner we learn that, the better for us all. Erik Erikson. At its core, we look at the solitary bee rehousing operation. Bees take up the space they need and work in tandem with other species through their daily activities. Humans are not so different. If we want to bring humans back in conversation with natural cycles, we can see our habitat being placed in a reimagined grocery store called the Bee Mart, 20 years into the future. A space that not only acts as a distribution center for products, but shifts our exposure of produce from simply retail to mutually benefiting preservation. This mart is a world-build element designed specifically to rethink the possibilities of urban connection to our foodways, with hopes of incorporating the same values we built the bee habitat with. At the center is our bee house, a carefully maintained pollinator mycelium hub. It is positioned as a hero piece, reminding people of their necessity and beauty. Around it is an immunity garden, serving plots for roots like ginger and shrubs like blueberries, harvestable natural foods specifically targeting immune health. The walls are vertical hydroponics, growing crops that are sold directly to the consumer. There are also collection facilities for household compost and fruits harvested from backyard trees, thus redistributing both waste and surplus food in a way that can be used by the community. Our most important jobs in this industry are growers, knowledgeable in cultivating plants, maintaining the bee habitat, and able to teach this information back to anybody looking to participate in our system. Different iterations of the structure are easily replicable at low costs, benefited by the lightweight and malleable nature of our material, placing them in backyards, schools, and gardens to form an underground network. When we imagine design with living organisms, we can create beyond existing life to support future generations. Time is a cycle of successive generations beyond an individual's life and death within an ecological context. Bees and every living thing operate with psychologist Erik Erikson's definition of generativity, a concern for establishing and guiding the next generation. It's time humans did this too. This generative life cycle model has inspired our design to participate in multiple influential life cycles involved with bees, fungi, crops, and our own. Through generations, we can revitalize these ecosystems from the ground up, providing natural immunity pathways for future life to grow on.
Awesome. Great job, University of Southern California. Let's start with Justin. Please, go ahead.
Thank you. That was incredible, guys. I think the storytelling is really good, and the concept and the research. I loved seeing the prototype. I think it's really clever. So well done. I'd love to know, have you done any research, or do you know why native bees aren't being used yet? I'm just wondering why, or is it just a kind of system thing? They just go with what's kind of the system is built for. Do you know, or is there not really any knowledge about that?
Well, I think one thing would be like honeybees have their social bees, so they're hives and it can be easily contained and kind of like multiplied. And solitary bees, it's harder to make them all work together, like for the purpose of like carrying them around and things like that, maybe.
Also with, industrial agriculture, there's been, like a monoculture in the crops that we were growing. And, the habitat fragmentation of not only the native bees, but the native plants that support the bees. So the solitary bee population has been declining for a long time.
Thank you so much. That's really great.
Bill, please, and then we'll go to Doug after.
Thank you for that wonderful presentation. I was wondering, in your renderings, you have the fruiting body of the mushroom growing. Is your vision that this will be dried and maybe a longer term installation, or will it be one that will have to be replenished? Because if the fruiting body grows, it will kind of consume all those materials.
Yeah, generally we were thinking about, having it open to different options. So the way that we built the prototype was with the 3 different parts because of the molds that we wanted to make. So it's possible that we could, do a combination of different things, but, sometimes we were thinking, okay, you can bake the mycelium with heat or just the sun so that it can harden and it will be more of a permanent structure. So we were thinking, it's possible to do that for maybe the top one and leave the middle one a little half baked, a little more sturdy, but still able to grow. And then the bottom would be untouched so that it can continue growing underneath the ground. So we were thinking, a combination of these things can be used as needed. Some structures may be more permanent, some may be more focused on growing the mycelium rather than providing like a long-term shelter for the bees. We'd like to, say it's possible for any of these versions.
Awesome. Let's finish up with Doug. Please go ahead.
Hi, I'm also enthusiastic about this project and excited to see it deployed. It's solving a problem that we normally think we can't solve, namely bee population collapse. I wonder, have you given any thought to giving feedback about how well the project is working? Like, is there any indication other than the fact that the hero pieces might decay, or you might see more bees? Thank you.
So is this about like the prototype that we've made, or hypothetically? Right now it's placed in like my backyard, and we haven't like buried it in the ground, but I have seen like flying insects enter. And I placed it next to a rosemary shrub, but I haven't really kept like notes or exactly on how many bees are in there currently.
That would be a great next step for us to work on.
Yeah.
Thank you.
Okay, cool. For the sake of time, we're going to move on to the next team. Thank you so much, University of Southern California. Great job. Next up, we have St. Francis College. Roll the tape.
As a teenager, I was very excited to get my nails done on a regular basis. That was until I had my first environmental science class, and since then, and in light of this 2022 Biodesign Challenge, Team Nails has been collaborating to create a natural acrylic alternative that is to reinvent a generational bonding experience and not only allows you to enjoy what your nails look like, but to also explore a fresh new take on luxury sustainability.
Nails are expressive. They're a form of expression. I went to Catholic school, and nails for us were a way of being expressive and showing individuality in a setting where those values weren't prized or encouraged. It was a way of literally and figuratively subverting uniformity.
But there's a dark side.
Focusing our efforts on the American market and more specifically targeting our own location, research yielded that some of the greatest costs associated with the nail industry are human costs. There has been extensive reporting on the dire conditions in which workers are forced to labor and the damages to their health are caused by the use of chemicals, adhesives, and solvents.
Traditional artificial nails are made from petroleum-based abs, acrylonitrile, butadiene, styrene, the same plastic used in Lego blocks. The lifetime of press-on nail after it has been used is more than 500 years.
The artificial nails market is projected to grow by $262.25 million between 2022 and 2026. Without an alternative, what would all that plastic mean? Assuming a $10 cost per package and 2-ounce weight per set, that's 1,504 metric tons of new plastic waste above current levels. Can we fix it? We asked over 30 people, including college students, salon owners, retail managers, people who prefer salon manicures, those who do their manicures at home, and of course, people who do their kids' nails when they ask. We asked these people who experience the manicure culture in very different ways, if a biodegradable option was available, would you use it? The response was overwhelming. Every single person we surveyed said yes.
We've invented nails, a biodegradable alternative to press-on nails.
Our research found only one biodegradable option for acrylic nails, and these are targeted at the in-salon gel manicure market, not everyday consumers.
Use the nail shapes as molds. Next, we use a recipe of 1/4 cup of water, a tablespoon of glycerol, half a tablespoon of cornstarch, and 1 cap of vinegar to create our biodegradable alternative. We heated it, let it cool, and poured it into our nail mold. After allowing the prototypes to cool for 1 to 3 days, we began our testing phase. We wanted to make sure that the nail would withstand the ingredients found in standard nail polish and lots of hand washing.
At the time of this recording, we have not tested for alcohol-based sanitizer or salt water, but we'll be conducting that testing shortly.
Nails should be as fun and as inexpensive as they are for my little sister. We should not have to sacrifice our self-care, healthy dose of vanity, or our creativity in exchange for doing the right thing by our environment.
We created this product with the hope of contributing, even in a small way, to both the need for accessible self-care alternatives and conscious alternatives that will neither break the bank or our planet.
Awesome. Thank you, St. Francis College. I appreciate the thought you put into that. Judges, do we have any questions? Yes, Todd, please.
Great project. This was really sort of interesting and something I've never really thought about, to be honest with you. So I really like you charging my brain and thinking in new ways. I'm curious about sort of some of the tests you ran on some of your nails, and were the nails you produced or able to produce kind of standing up to the nails that you would buy in the store, and what are your kind of next steps to test one for the nail polishes, but then also I was wondering if you have sort of ideas in terms of dealing with that other problem, which is sort of the nail polish side of this as well.
Yeah, so basically to start, we really only first tested with normal hand soap and warm water. It worked well. It didn't dissolve, but we have next steps to focus on the adhesive part of the nail. And nail polish, we really just focused on the base of the actual alternative to plastic, so the nail itself. We do have future sorry, we do have future plans for this project. We're in the midst of applying to a grant at our school, which is called the St. Francis College Center for Entrepreneurship. So we're going to apply for a grant where we can get funding to develop this project even more. And we're also focusing on even looking into a sustainable option for the nail polish itself, as well as the nail that we've been working on.
Yeah, and to add, we also did some alcohol testing on like regular hand sanitizer kind of test, and it was able to last during that time too, but we didn't do a full day of testing. We only did about half a day of testing. So in that realm, we don't really know. But when it came to the enamel that was added by the nail polish on top of that plastic nail, it did aid probably in the durability of that and the long lasting of it.
So I'm sorry, can you hear me? Oh my God. Okay. So just to kind of wrap it all into a bow, we identified several points of toxicity within the nail industry. So we talked about the solvents, we talked about the adhesives, we talked about the nail polishes. So one of the reasons why we focused on the nail itself is because if you have an in-salon experience, that is the foundation of that experience. First, they would add that nail, and then they would obviously use an adhesive. They would add the nail polishes and different gels and polymers to that in order to form up the nail or to create the nail art. So we started at the basic foundation, but we do plan to potentially move into the other areas where we saw the problems.
Awesome. Great answers. Justin, please go ahead.
Yeah, really great project, guys. When I saw the description, I was really excited about it as someone who does enjoy the nail culture. And I think it's definitely a huge problem to address. And if you can solve it in some way, that would be huge. So this is a really big problem space and relevant and important. I was really impressed by the level of testing you guys did as a first attempt and kind of first iterations. It's really exciting to think about where you might go with some more funding and time and research. So I think you're at a great point for a first iteration. My question is, where did you start with trying to come up with the recipe? I thought like the recipe was really interesting and it's obviously very home-based kind of ingredients, but how did you even land on that recipe?
So basically we were in a biodesign class together. So we were looking at different options that we can create something that's biodegradable, something that's something that we have in our home. And basically we came to the ingredients, because, well, during COVID, we're all at home, you know what I mean? So we use things in our house that we don't really realize that we have. And I realized that doing more research into biology and biodesign, I realized that bioplastic is a really not common thing. It's something that's becoming. And when I looked into different ways of how you can create bioplastic and look into it, I kind of looked into what I had in my house, and I had glycerol, I had cornstarch that you use for baking, some vinegar, and I really did a few different combinations of ingredients to create one of the prototypes.
Great, thank you. So you worked with a bioplastic kind of recipe. Sorry, Josephina, go for it if you want to add. That's great, though. Thank you.
Sorry about that. I just wanted to add that. So as part of this class, we each individually were working on different types of bioplastics to solve different problems. And so we individually had different recipes. And so it was about, at some point, we came together as a group and decided to move forward with one idea, but we already had this background where all three of us had tested different formulas.
That's great. Thank you so much.
Yeah, and we also got some examples off of the Materium website to find out about possible ingredients that we could use, and the Materium website was super helpful with that.
Awesome. Okay, one more question from Anya. Please go ahead.
I like how you guys found a nice little niche that you could, make a little bit more sustainable. I'm curious, from somebody who obviously doesn't go to the nail salon to get acrylic nails. Why would somebody maybe not want to use your product? I know it's kind of like a little bit different way of approaching things, but why would they, maybe opt for the more conventional approach over what you're doing?
I could answer that, I think. So for two reasons. The first reason is it's really more cultural, right? We become accustomed to whatever the status quo is. So there might be questions as to the durability of our product versus what is currently on the market. And the other thing is this perception of cost, right? So we always think that the more biodegradable option, the more ecological option, the more organic option in terms of food is going to be more expensive. So one of the aims that we have for this product is to maintain its accessibility for everybody.
Thanks. Good product.
Awesome. Thank you, judges. Thank you, team. Great job. We're going to move on to our last video of the afternoon, the evening, wherever you are in the world. University of the Sunshine Coast.
Hello, I'm Millie. I'm Jess. I'm Keely. And I'm Lily. And we're Mid-Tide. We're a group of second-year graphic design students from the University of the Sunshine Coast in Queensland, Australia. We have developed a product concept for a sustainable and reef-safe sunscreen. We've employed biomimicry to help reduce the effects of climate change and coral bleaching. We chose this global issue as locals of the Australian coastline. We are passionate about protecting our home and the environment so we can continue to do the things we love in the ocean.
Coral bleaching occurs when corals are stressed by altered environmental conditions. This includes changes in temperature, light, or nutrients, causing the algae that lives in their tissues to be expelled, resulting in a white or bleached appearance. It is also estimated that 10% of coral reefs worldwide are threatened by sunscreen-induced bleaching. Mid-Tide is a small-scale initiative that aims to forge new sun and reef protection technology. This new product utilizes biomimicry to emulate the elements and structures found within our natural environment. Mid-Tide sunscreen is unique, as we only use natural environmentally friendly ingredients and fully biodegradable packaging. Our revolutionary product will benefit those who wear our sunscreen and the environment. Currently, there are many sources of water pollutants finding their way into our waterways, such as fertilizers, plastics, oils, and sunscreens. It is estimated that between 4,000 to 6,000 metric tons of sunscreen wash off beachgoers annually, whilst the residue on our skin is washed into waterways through showers. Currently, more than 70% of sunscreens on the market contain harmful chemicals that have been banned in some locations around the globe.
Our sunscreen employs biomimicry, which is a way of solving issues through observing and imitating the environment around us. For our sunscreen, we looked at organisms that have natural UV protection, such as the zebrafish, hippopotamuses, and some sea urchin species. These organisms synthesize a compound called gadrosol, which offers them natural UV protection. Similarly, we looked at palathine, which is a UV protectant that seaweed creates as a chemical barrier. We aim to not only include gadrosol and palathine in our formula, but also restorative ingredients such as calcium carbonate. As previously mentioned, the amount of sunscreen that washes off our skin is extraordinary, so we decided to use this fact to our advantage and use calcium carbonate. Calcium carbonate has been tested as a UV filter on the water surface and has proven to be a promising method for reducing the effects of UV rays on reefs. Furthermore, corals require adequate calcium levels to deposit the structures they live in, so including it in our formula was essential.
Not only are chemicals harming our vulnerable marine environments, microplastics are also causing havoc. To produce a more sustainable product and eliminate the introduction of more microplastics into our oceans, we will use a specific plastic called high-density polyethylene, HDPE. This plastic is 100% biodegradable and a recyclable option which is able to degrade without leaving behind microplastics. Furthermore, this specific type of plastic will degrade in a normal landfill in less than 10 years, unlike other plastics which can take hundreds of years to degrade and still leave behind microplastics. We have created Mid-Tide with the intention of a multi-beneficial product that protects humans and the reefs. Using seaweed technology and additional ingredients such as calcium carbonate, all have the potential to act as nutrient supplements with the concept of a regenerative product. Biomimicry-based solutions are the future of climate-conscious design, something we believe to be a foundation in solving some of our global wicked problems. While reef-safe sunscreens are on the market and are highly supported in places such as Hawaii, there are not currently any regulations for reef-safe products, nor are they widely available or supported in the Australian market. Once established, Mid-Tide would cycle our profits back into biomimicry innovation, research and protection funding for the reefs, starting with the world's largest, the Great Barrier Reef. Creating a new reef-safe and restorative sunscreen is something that is small, realistic, and is an easy-to-implement solution to protect your skin, whilst also protecting the reef.
Throughout the course of this Biodesign challenge, our team has come together on relatively short notice to produce a multimedia presentation and well-thought-out product. As graphic design students, we've really developed our brand and been able to obtain new skills throughout the challenge. Not only this, but as a team, we've supported each other and allowed each other to really demonstrate our personal strengths. Considering the fact that we are graphic design students and don't necessarily have a background in science, our research has come from secondary sources. So the research side is definitely something that we would love to have been more involved with, particularly if we were to take this project further. When considering the logistics of our product, it would need to be used in combination with other solutions, such as education and real legislative change. For us as a group, we've all grown up within 30 minutes of the beach and we really value the health of the environment. It's important to remember that reefs are valuable for a number of other reasons. The Great Barrier Reef, for example, it's a heritage-listed site which has cultural significance and is also a huge tourism attraction. Our reefs are imperative to a range of stakeholders. We would also value the input of our local Indigenous Kabi Kabi community, ecologists and biologists, to see how we could work together to protect these sacred areas that are important to us all. We believe these are important perspectives to consider.
We have learned a lot about biodesign through the development of this project and have really enjoyed deepening our knowledge on design features. Thank you for listening to our biodesign concept for Mid-Tide.
Awesome. Wonderful job, team. Judges, do we have any questions for Melly? Yeah, Phil, please. Take it away.
Thank you for that presentation. I was wondering if there was, you did mention policy and how they're different in different parts of the world. In Queensland, is there any key policy that I think you would identify as something that needs to change in order to help people accept these kind of products?
Definitely. I think that there should be more regulation on these harmful ingredients, especially when there are alternatives. Zinc oxide-based sunscreens have become more popular, especially in our area, but those mainstream sunscreen brands that use these harmful ingredients are still way more popular in the shops, a lot more accessible and a lot cheaper. Sunscreen here is really expensive. Considering the damage that the sun does here, we have a really high UV factor. So it's pretty shocking that there isn't more regulation done to protect us and our environment here.
Thanks, Phil. Judges, any other questions? Todd, please.
I guess I maybe have two questions. One is obviously the sunscreen has to do its job, right, in terms of blocking the UV rays. So I was curious in terms of your thinking of moving forward, how you would go about or plan on, testing or thinking about whether or not this biomimicry will actually do what it needs to do on humans or on human skin.
Yeah, so there's a few things we've looked at. There is the seaweed research group at USC, and that's made up of more than 30 professionals, students, mentors, PhD students. And they've indicated that they're interested to move forward with us, kind of basing off the technology that they've already, established. And that's for both the packaging and the ingredients themselves. But in terms of more of the biomimicry on getting the Gadrisol and Palathine ingredients. We have looked at a company called Gadrisol Laboratories, and they're doing a lot of extensive research into how to actually get these ingredients that are amino acids that we don't already produce but are found in nature, and how that can be used for sunscreen. So there isn't really a product yet. We've kind of developed more like the face of a brand that could use those technologies. But we're definitely looking into how we can work with some of these companies to move forward with some prototypes and research.
Todd, did you have a second question? Or you feel good?
I always have lots of questions, so if other people have questions, like, go ahead.
Judges, I'll keep the space open for anyone that has another question.
I can throw one in. Naively, I might think that sunscreen was good for the coral reef, right? Because it washes off my skin and gets into the water and actually keeps the UV rays from hitting the coral reef, right? I know this is not the case at all, but one of the points that you bring up is that we could make sunscreen that's actually beneficial to the reef. How do you think you might educate us about that and convince us that your solution is better?
Yeah, so we're a group of 4 graphic design students. So that's actually the path we took further. Not all of the stuff we've created is actually in that video, but we created some fact sheets and stuff that we worked with a program called iJack to animate. So we were looking at branding that uses like virtual reality and AR to kind of bring customers into that experience. So instead of it being, research when it would go to the public, it's more about immersive and actually family-friendly product that can kind of show the products you may have used in the past and relied on to keep you safe may actually be doing more harm than good, not only for the reefs, but also potentially for your own skin internally with the absorption and everything. So it's more just that kind of branding that we've established that we think could be really good in the future.
Thanks.
Thanks, Doug. Eve, please go ahead.
Yeah, in terms of your reference for in terms of biomimicry, your natural references, did you look into have you considered, because you mentioned the effects of the current sunscreen on the coral, for example, right? But did you consider like what the natural references would, like what effects it would have on to the coral reefs? Does that make the question make sense?
As in, like the ingredients we've used, what effect they could have on the reefs?
Yeah.
Yeah. So we'd have to do a lot of testing to see that it is safe. Obviously, if you make a reef safe sunscreen, your claims need to be accurate, which is kind of one of the issues that a lot of companies have, is that they claim these things and they're maybe not actually regulated to be true. So the Gadrisol and Palathine concepts, we don't fully understand how they would work with coral reefs, but because we've seen that seaweed actually creates their own UV protection, Palathine, and then coral also has UV protection in it that it develops. The kind of thinking we had behind it is that it should be safe. We would have to test that. Whereas the calcium carbonate is an ingredient, a nutrient that coral already creates itself. And that is fundamental in keeping it healthy. So having that as an ingredient in there has that regenerative properties. We would have to test it to make sure these are correct and definitely right when combined. But the idea is that it's a product that potentially could actually reverse some damage that we have been doing to the reefs and make it a healthier environment for them.
Thank you.
Wonderful. Thank you, Millie. I'm excited to see where you take this next. We're at the end for today. So Alex, could I ask you to join us on video, please?
Hi there.
Oh, there you are. Okay, cool. Great job to the students for today. That was a long day, but we got through it, and we got some wonderful presentations and questions and answers. We thank you all in the audience for joining us today. Thank you to our judges for all of your questions. Our final day for student presentation is tomorrow. The day will begin with presentations at 8 a.m. Eastern time, and then we'll end actually with a gallery show in the afternoon, all ET. So the gallery show is running from 2:30 to 6 p.m. Eastern time, and it's going to be taking place on Zoom. So we ask you all to join us, judges, instructors, students, everyone in our community out there in the audience. It's a great place for you all to meet one another, to speak one-on-one, to make new collaborations, to find new people to work with. So we hope you'll join us for that.
And we mentioned this earlier in the day. If you're just joining us, please do cast a vote in our Community Choice Prize award. It is open to anyone in the world. So I know we're a super international crowd this year. You can vote from the link on our BDC Summit page. We have almost 500 votes now, and there isn't a clear winner, so it's anyone's win. Voting will end at 12 o'clock on Thursday, June 23rd. And I was going to say may the best team win, but may a team win. You're all the best. All right. And with that, thank you so much, everyone. It's been a pleasure hearing all the presentations and the feedback from the judges, and can't wait for tomorrow for more.
Yeah. Have a great rest of your day, everybody. Take care.
Thank you.